So I'm just going to give you an idea about what you can do with, what kind of experiments that you can do in a clinical setting, and how we have actually used this in our work, right? So I've titled my talk, How You Can Use Transcriptomics in Clinical Research. So I mean, right now I don't think we need to talk about this. So we are basically going to be concentrating on just this part, the transcriptomics. And so I mean, again, I think you're all now well versed with what is transcriptome, right? So I'm just going to skip this. But the only thing to remember here is that a transcriptome is a temporary state, right? It's a snapshot at one given time point. So it is not going to represent the cell at all the stages, right? So but in a bulk thing, you will have cells at different cycles, I mean at different stages of the development, but they'll all be fixed at one time, right? So it's a snapshot of that time. So that is one thing that you have to remember. And of course, there are different kinds of RNA, and depending upon the work that you design, you can have different libraries, and you can actually quantify all these kinds as per your study protocols. So the only reason that I've put up this timeline is to just show you that from the invention of the PCR in '84, it took actually a decade to come up with the real-time PCR. I mean, that is how long it took. And now in 2016, spatial transcriptomics was in the picture. So after that, development was really fast after the advent of next generation sequencing. So there are different kinds. My talk is basically about this. And tomorrow we will talk about the single cell RNA sequencing. And probably if we have time, they can tell us a bit about the spatial transcriptomics as well, which is probably of more interest to the pathologist, right? So different kinds of RNA sequencing platforms are available, and I'll be talking more about what we have used, which is the standard short-read RNA-seq as well as the small RNAs. And before I tell you all that you can do, I'll tell you a bit about the different repositories. So this is important because the moment you generate some data, it has to be shared. So there are different repositories that are available where you have to upload your all the data that you generate. So like commonly we use the gene expression omnibus. So you have to upload your data, it is checked by the curators there, and an accession number is provided. And only then you can actually publish your data, and you can decide when you would want this data to be made available to the public, and other people can actually download it and use it for their own analysis as well. So these are some other resources that are available, and you can probably explore them. So yeah, I mean by now you all know all this really well. So these are the different steps, different workflow in RNA-seq, and Francesco is kind of taking you through this, and he'll be talking about the next steps today and tomorrow as well. So I'm not going to talk about this, but what I do want to show you is a very simple experimental design. What we did, that's how we actually started, this work. So we had a disease condition and a healthy condition, and we really wanted to see what's the difference. Very simple question. I'm not going to talk about all the different factors that weighed in, but this was the basic idea. And again, after doing the simple steps of collecting the sample, which is actually not simple, there are many nuances to it, but I'll skip that. We can probably discuss that in some other day. I mean even, so even actually what kind of kits that you would use, what is the order of sample draw? That is also really important if you have to go for an RNA sequencing experiment. All these things will matter. So these are the certain things which you have to keep in mind before you design your experiment. And then you have your data which comes out. You have these different steps, and probably we are now at the HiSat2 stage. We are aligning our data to the reference, and we'll be using feature counts and dseq2. This we'll do today. So again, once you start doing your own work, this is just a brief about how it will look on your systems, right? So this is a snapshot when we are running the fast QC, the Trimomatic analysis, and HiSat2. So you're already doing this, so that's why I'm kind of not going into the details. So eventually you will have your dseq2, which you will be, now after this step you have your differentially expressed genes. So now if you remember what Dr. Boni did yesterday, he took you through a very simple way to plot your data in R, and he actually arrived at this very beautiful plot, right? So there was a reason why he took you from that. So now I think most of us can do this. You will have to do this here because the data is huge. It's not a couple of 100 reads that you will get, it will be in thousands and lakhs, right? So you cannot manipulate it with the normal Excel or something like that. You have to do it in R. So once you have your data, now by using, remember, so we had put some, we had used the p-values and we had decided that I want to see the data or the DEGs which are above a certain cutoff or below a certain cutoff. So that is precisely what I've done here. The most highly up-regulated and down-regulated genes are highlighted here, and you can also have the names show up. So all of this can be done pretty easily because now you have the background, right? So once you have done this, you can actually, after this comes your visualizations. So how do you see your data? That's some part of it I think Dr. Boni will take tomorrow, but I'll show you what we did for a very simple experimental design. So I feel that, you know, transcriptomics can be used in multiple ways. So some of this, which are I think specific for our unique niche, would be disease-specific biomarker discovery. I think a lot of people in PGI are working on this. Disease diagnosis and monitoring. I'll show you some examples where we've done this. And understanding the disease mechanism. So precision medicine is something I have no experience in, but probably our other departments might be using this as well. And obviously, finally, for guideline development standardization. But that's a far way to go for us at least. So I'll just now give you one example where we've used transcriptomics to look at the disease pathogenesis. So our unit is allergy immunology unit. We work in the area of primary immunodeficiencies. Now they are called as inborn errors of immunity. So most of these have a monogenic cause ascribed to them. But a disease like CVID, common variable immunodeficiency, the genetic, there is no one monogenic cause which has been described. So in this case, to understand why a patient presents with a deranged immune profile is interesting. So for this, we've been looking at this disease at different angles, but I'm just going to talk about the transcriptomic part today. So very simple experiment design. We have a group of patients, we have healthy controls. So we just simply wanted to see what's happening in the neutrophils or in the innate cells as per your design. So again, simply collect your samples, do your RNA-seq, get the data, and then comes a visualization. So this is both PBMCs and neutrophils. So I'm just giving you just a snapshot of how your data would look like. It depends on your experimental design. What is your research question? So here in this case, so again, I could see that there are a lot of genes which are significantly upregulated and downregulated. So it depends on what is your criteria. How stringent do you want it to be, or how relaxed do you want it to be? So those are the parameters you can play with and see if it makes sense for you. Again, these are very good looking plots, good for the reviewers, but you need to understand what will you do with it. There's a huge amount of information that is available now. So again, you will have an idea about the genes which are upregulated and downregulated in a certain condition and the common ones. So from there, you can start building up your story. So you can see the function of these genes. For this, we have some different tools, but you can even start searching yourself. If you understand your disease, so again, you will have to revisit the literature, understand what is the pathogenesis, and then correlate with the data that you generate. So again, once you have your differential counts, right, expression counts, so you can actually go ahead and start making these plots. So this is called the gene ontology enrichment analysis. So what does this mean? So this picture actually gives you a lot of information. So what you can see is you can actually look at the different processes, where this is localize the cellular compartment, the biological process, and the molecular function as well. So the color of the bubble denotes the process type. The intensity is how significantly it is either up or down regulated, depends on how you've plotted it, right? And finally, this bigger the bubble, the more are the number of genes in that particular geo process, right? So if it is a bigger bubble, so more genes. So by this, in one graph, you can have all these processes which you can actually see. Like for example, for me in this disease, I'm more interested in the immune responses. So can you see here? So regulation of immune response. Some of these hits are pretty highly significant, around 40 gene sets are present here. So now you have an idea, okay, these are the processes which are important. Then you can move ahead and there are many other visualizations. I'm just giving you a very brief overview. So you can do your path view analysis. In this, you can actually superimpose the genes which are up or down regulated these are all different pathways. Like for example, I chose this P53 signaling pathway. So you can see some which are in red. You can decide, right? For me, I've taken red to be upregulated, green to be down. It's up to you. So now you can very nicely see the data that you get, actually what is happening. So in a particular pathway or in a particular, for example, the B cell receptor signaling pathway, what is happening here? So I mean, for us working in a clinical setup, this would make sense, right? I mean, when we see this, we can actually interpret and correlate, like, you know, this is my patient and eventually this is happening. So now I know that in CVID patient, something is messed up with the B cell receptor signaling pathway. Actually, you will see a lot of hits here just for today's presentation, just not to make it too colorful. I mean, not to overwhelm everyone. So I've just kept these 1 or 2 genes just to show you. You will have this kind of analysis that you can actually do with your transcriptome data. So basically for us, you know, we got a lot of insights from this data. We saw that there was some immune dysregulation happening in our patients. We could find out that apoptosis and cell cytoregulation was impaired. So you know, these are the kind of insights which you can get, and finally you can take this work forward.
These were made on p-value or adjusted p-value?
Adjusted p-value. I mean, again, we'll just see it in the examples today, and some of these visualizations Dr. Boni will take tomorrow. So you can just, I can show you some tools also that you can do online. There are some online tools also available. Just put your gene set list and it will show you these pathways. So we can go through that as well. So, a bit about how you can go for disease-specific biomarker discovery. This is another work that we did in Kawasaki disease. So this is now the commonest cause of acquired childhood heart disease in India. And in this, you know, we wanted to see if we can use our data sets and the publicly available data sets and see if we can find some potential biomarkers which we can further evaluate in a larger cohort. So for this, we had downloaded data, and then, you know, we had our own data. So we tried to see any commonality between the genes. So we found a couple of good hits. These genes which were found to be involved in neutrophil degranulation, they were you know, highly expressed in patients with Kawasaki disease. So this is something that we are actually following up in our lab. So again, this is how you can even actually use publicly available database, and you can, you know, take your own data, see if you find some commonality or some differences, and you can design some kind of a study. I feel this is something which we really do a lot in PGI. So, you know, rather than going the old, I mean, not the old fashioned, the regular way by going through the proteome, you can even start here. That is also fine, but that's another way to look at things, right? If you have a very volatile disease condition, something like that. So a bit about disease diagnosis and monitoring. This was something have you heard about this? How many read about this? Yeah, I think most of us, right? This was really, you know, something happening so many headlines. Oh, black fungus, people are dying. You know, this was during the 2021 Delta wave of the COVID. We got a lot of upsurgence of mucormycosis cases, especially in India, along with aspergillosis. So we wanted to see that what is happening. So amongst the many different things that we were evaluating, we also kind of thought about looking at how the immune cells are responding. So what is different in these patients as compared to patients who present with only COVID? The healthy patients who we assume who were negative for the COVID PCR. So that was one group. One group is the COVID people who were positive for the COVID PCR. One group who presented with just mucor infection and no COVID, and the other group was the COVID and mucor, right? So this is a lot of factors here. We'll also talk about that later. So that study, and we were looking at the different compartments, the innate compartment and the adaptive compartment as well. So what we did was, I've just told you the methodology, and again we did all those analyses, and we finally found some pathways which were really upregulated in these patients, especially netosis. This is something, we would assume that this is something that you would see in pathology, but the moment you fix your tissue, the nets get degraded. So you will not actually see them. So here in the case, you can see all the genes which are involved in neutrophil degranulation, which eventually leads to netosis, were highly upregulated. If you can see, most of these are red, right? So that means in these patients, this was really something which was happening a lot. And it actually matches with the clinical phenotype that we were observing in these patients. So I'll not talk about this. And I mean, of course, by this you can actually study the disease mechanism. And this is about, so we also wanted to look at what is the disease progression? How does COVID-19 lead to mucormycosis, or does it there is a predisposition in these patients, or what is it? So this is some kind of analysis we are still in the process of doing. We've not really come to a conclusion, but the reason I show you this is that if you have like a disease progression, something like this can also be done, you know. You have at N0 stage, N1, N2, you can have different snapshots, and you can actually compare and see how your disease or process is evolving. Uh, Finally, I'll just one or two slides more, and we can go to the hands-on. So in this case, we also have a bit of a microbiology, because that's what I've trained in, can't let it go. So we've also looked at the biofilm. So there's a couple of papers which we published here, and we wanted to see the contribution of alternative transcript isoforms in the biofilms. So this is one work from where this entire story started. So we actually did not stop at just the gene ontology. We went ahead and looked at the isoform also, because this would really be important, especially in eukaryotes, right? Because we have alternative splicing. So different isoforms are generated. So what is the isoform usage? So if you just see, I'll just take 2 minutes. You can look at the gene expression in the healthy versus the disease condition. Then you will have the isoform expression. What do I mean by this? These are the different isoforms of one gene, and in the diseased versus the healthy condition, you can see there is some difference, right? And then you can actually see which isoform is being used at that point. So this is called as the isoform usage. So out of all the different isoforms, this is the only significant one. So, but then we did not stop here, we took it a step further, and we wanted to see how these isoforms will contribute to the different domain architecture. So this is an example of nuclear factor kappa beta 1. So now I can see that my complete protein would have all these domains. So this is the one protein which is, this is a transcript which is actually encoding for all these proteins. But is this the one which is highly upregulated or expressed, or the one which is used? And this is how you basically can actually see if the isoform which is significantly being expressed in your condition, is it the one which is forming these different proteins, right? So again, this is something which can give you a lot of clues about, is it, if you're not sure about the disease pathogenesis, this can be a major factor, especially in infectious diseases, that, you know, the pathogen can subvert the human response and, you know, change the isoform usage, make the protein which is convenient to the pathogen. So that is an upcoming mechanism which is being discussed a lot. So I'll skip with this and finally leave you with this thought, would you like to seek or not? So thank you, everyone. Mistakes.
Yes. Okay, good. My best friend. Good morning. I'm still a bit sleepy, I think. So if I talk too slow or you see I'm falling asleep, let me know. Just scream. Well, the goals for today, right? So today you're going to be working in R, finally, I guess. So the main goal today is going to be, we're going to do differential gene expression analysis using this R package called DSeq2. Again, we were going to do it hands-on. That's the main idea. But before that, I'm going to review some of the concepts related to DSeq2. This is what we saw yesterday, right? So yesterday, what we did was we went from the QCM filtering of our FASTQ files. We aligned it to the genome. And we use a string type plus that a small tool to create our count table, our raw count table, or the count matrix, we can call it too. So today we're going to focus on differential gene expression analysis. And again, depending on the time we have, I don't know if it's going to be today or maybe tomorrow, we're going to use it. We're going to do pathway analysis maybe with this data or with the single cell data that we're going to analyze tomorrow. This is how we finished yesterday. This is our count table, do you remember? Here you go, our raw count table matrix. The samples we did yesterday were just a test. Today, what I did is I got the actual full set of samples, and I processed them. And in your folders, when we log into our Jupyter app, you're going to have full count matrix. So for differential gene expression, we have different options. I think I already mentioned that yesterday. We're going to use DSeq2. I would say that's probably one of the most popular ones, if not the most, with the goal of producing some tables with the differential gene expression analysis, log2 changes, p-values, q-values. How many of you have seen one of those final tables with the gene counts, p-values, 1, 2? Oh, okay, more. So you're a bit familiar with it. Okay, so DSeq2, it's part of I'm going to just show it. It's part of the bioconductor. Well, how many people have heard about bioconductor or use R with bioconductor? A couple, okay, a little bit less. So in bioconductor, it's like a database. It's a repository of R packages, all related to bioinformatics. And DSeq is part of it. So bioconductor is well documented. There's a lot of information there. So I recommend you, if you're going to do analysis, you can always consult what is available there for you. It's all related to R, though. The advantage of DSeq2, so it allows you to create from very simple models to analyze your data for more complex models like, oh, I want to remove batch effects, or the example that I gave you yesterday that included not only one source of variability in your data, the condition, but there were other sources like genotype or I don't know what else it was, the sex of the animal, right? All those kind of things. You can create relatively complex models and still do an analysis with your data. It's not just, oh, I'm comparing wild type versus control. You can go farther than that. Well, this is just the paper, the original paper for DSeq2. Okay. To start the analysis with DSeq2, you need 3 pieces of information. As I said, your count matrix. You're going to need what we call the metadata. We talked about this yesterday too. So the information that you have about of the sample, right? Generally, is going to be the simple example is going to be you're going to have a few samples that are your control and a few samples that you treated, or there's something special about it that you want to compare with it. These metadata can be more complex than this. Again, going back to the example of the mice being in cages and the sex and the genotype, you can create bigger metadata files. And you need your You need a pointer. That's where you need a better. And you need your design, right? So in this example, we'll be pretty simple. We are expecting that the condition that we are studying is the main responsible for the gene expression differences. And this is how we're going to express it in DSeq2. And I've been showing examples of this, right? You recognize that tilde here and how you express the formula after it. It's the same way that the linear models in R express the formulas, like for linear regression or anything else you do in those simple linear or generalized linear models. Here is the, right. So if you would do a linear regression, it would look like this, right? So in the formula, we skip the intercept or the baseline to just give the other parameters. But as I was mentioning, you can even build more complex models. And if we have time, we'll try to do an example with this. In the practical and the hands-on practical, we're going to start with the simple one. But if we have time, we'll go something as complex as this if we can. OK, so if you are in R, the only two commands that you need to do your analysis initially just would be these two, nothing else. So in one, you're going to provide the metrics, the metadata, your design formula, and the DSeq2 call will do the analysis for you. And that's it. Pretty simple, right? Well, we're going to spend 2 hours just exploring the data probably and doing other plots and doing other things. But pretty much an analysis will consist in 2 lines. You can add a couple more to get the results, and that's pretty much it. So as I said, the count data, metadata, and your design. So when you run this, you're going to get some report that DSeq2 is doing some analysis. So we're going to talk about the normalization. The way that DSeq2 normalize the data, they call it, they use size factors. That's what DSeq2 call them. And I'll show you how you obtain them. I'm going to give some details about how you do that. Again, it's one of those things that you're going to see once, and then you're going to forget about it. You don't have to memorize how all the steps of the analysis are. But yeah, we need to do normalization. And the normalization in DSeq2 is by scaling the counts with a size factor, meaning we're going to divide every count that we have in our data metrics with a size factor per sample. It estimates the dispersion of the data, meaning how much noise you have and how much signal comes from the replicates. And then we're going to use the generalized linear models to calculate the fit to the data and calculate the fold changes. Or if I say coefficient sometimes, I mean fold changes too. They are interchange. So that would be our slope in our regression. For in this case, it's going to be our fold change. And then, of course, it's going to estimate our p-value. Basically, it's going to test our hypothesis, the null hypothesis that is our gene being differentially expressed or not. Is the fold change different from 0 or not? OK, why we need normalization, right? And we talk about this a little bit too. And you guys already mentioned, OK, yes, every sample is going to be sequenced at different depth. So we need to compensate for that, right? So if you have a sample that is deeper sequenced, that sample is going to get more reads. That's fairly logic, right? There are other things we can consider for normalization. One is the gene length, and the other one is the RNA composition. Sorry. Right, here I'm giving an example, right? So we have sample A and sample B. You can imagine that if you sequence 10 times more sample A, you're going to get more reads for sample A. That doesn't mean that it's differentially expressed, right? It's just you sequence more. The other thing is for a short gene, you're going to get less read than for a longer one. That is going to accumulate more reads across the gene length. And I'm going to talk about RNA composition later because it's the harder one to, let's say, understand. But I'm assuming you understand the basics of gene length and sequence length, right? Why we would want to normalize for them. Yep. So here I'm listing some of the common methods or methods and units of normalization. You probably have heard, or I don't know, I definitely, when I started doing transcriptomics, this is where the most common ones are PKMs and FPKMs. You still hear them, right? We use them less and less now because we see problems with them. But this is probably one of the most common ones we've had for the past 20 years. I said that for DSeq2 we use the size factor, but if you use another program like HR, it's going to use a different unit. If you notice, most of them already take into account the sequencing length, right? They use some sort of normalization like dividing by the total number of reads or using sort of the division by the total number of reads. So RPKNs, FPKNs, and TPMs, they use the sequencing length, or all of them use the sequencing depth, but these two also use the gene length. Why do you think maybe it's not that important to use the gene length of a gene for normalization? Or why DSeq2, for example, that is one of the most popular tools that doesn't use the length of a gene for normalizing? Any ideas? I'm asking why it's not important, or why it's not so important that DSeq2 decide not to use the length of a gene for normalizing. Well, basically you are comparing one gene across samples. The length of the gene doesn't change. It doesn't matter if it's sample A, sample B, or sample C. The gene length is the same, so you don't need to normalize for that, in theory, right? An exception would be if you want to compare within a sample across genes, but we don't do that, right? What we do is to compare the same gene across different samples, and the length of the gene doesn't change. So that is why that more modern approaches, let's say modern approaches, they don't use the gene length for normalization. They use another thing, which is the RNA composition. Well, but this is another example, right? So here I'm giving what is important to normalize for the sequencing length. We had sample 1 that is bit sequenced to this depth. If you double every single gene, you get the double of the sequencing depth. But it doesn't mean sample 2 it's 2 times more expressed than sample 1, right? It's just been 2 times more sequenced. Okay, so now I'm going to talk about the RNA composition, or what we also call library composition. So there can be situations where you compare, for example, heart versus lung, or 2 different tissues. And you may find that only some genes are expressed in heart, and they are 0 expressed in the other sample, right? So this is a type of what we call different RNA composition, right? You already know there are going to be a bunch of genes that are expressed in one and completely turned down in a different sample. Another one could be if you have a KO, right? So if you knocked out the transcription factor, all of a sudden everything that transcription factor controls is not going to be expressed in your KO compared with the control. I mean, you want those situations, right? You want to know what genes are differentially expressed, but that creates a problem. So in this case, I put a really simple example here, right, with a few genes. So I know it's hard to imagine that it is a situation like this, but let's imagine you have this sample. Both of them are exactly sequenced with the same number of sequences, right? But all of a sudden, you knock out one of the genes, or for whatever reason in this sample, the gene is not expressed. So you're going to have to distribute these 627 reads across all the other genes, right? Because both of them are sequenced at the same sequencing depth. So what's going to happen is you're going to have to distribute them, but actually these genes are not differentially expressed. If you try to do an analysis where the RNA composition is not taken into account, you're going to say, yes, this sample 2 for this gene has 4 times the expression level is higher, right? So the rest of the genes are going to look differentially expressed. Actually, if you do an analysis with such a small table where the RNA composition is not taken into account, all of them are being differentially expressed, which is not true. The only one is differentially expressed is the one that is knocked out. Yep, so that's what I'm trying to express with this simple example. What is the problem of RNA composition? When you have a bunch of genes that are knocked out, and you don't consider the problem of RNA composition in normalization. Okay, so this is the part where I'm going to go into a little bit into details. Again, you don't have to know exactly how it gets all the steps in the process. This is how DSeq2 normalizes, and I want to go through this a little bit just to show you how the RNA composition is taken into account in the normalization process. Basically, the first step is what is called getting the geometric mean for every row in your table. So basically, you just transform the every count in the natural log and then getting the mean. So that's the geometric mean, right? The advantage of using logs and everything else when you normalize is that you are removing the extreme cases, right? And focusing only on those genes that are, let's say, more normal express, right? The housekeeping genes and things like that. Another problem of doing the log transformation is, and in R, right, a 0, the log natural of 0, is going to be transformed to a minus infinitive, right? So DSeq2, what it does is removing genes that have a 0 count. So those genes are not taken into account when you do the normalization process. So meaning you are removing the RNA composition problem of genes that are only expressed in one sample and not in the others. Then you subtract the geometric mean to the counts, and it goes through this process. Again, you don't have to memorize all this. Then it goes for every sample. It calculates the mean of the ratios that you did. Now it goes back to denormalize. So it goes back to the natural number. So it transforms the log 2 to a natural number. And that is going to be your size factors of every sample, right? So you calculate a size factor for every sample. And the only thing you do when you have those size factors is divide your counts by that size factor. So what I'm trying to tell you is that doing all those steps of doing log transformations and the geometric means, you're trying to remove extremes outliers of counts that may make your normalization process be biased to very weird numbers that may not be representative of real signal. Again, the median of the factors also helps to just focusing on housekeeping genes. There's another thing, right? All this process where DSeq2 is doing is assuming that most of the genes are not differentially expressed, right? Because you are removing the ones that have 0 counts, so they are not taking into account those, but you know that's not going to be differentially expressed. You're also removing extreme cases that may be also differentially expressed. So it's making the assumption that most of the genes are not going to be differentially expressed. Another thing is that through this process, we've been using all the samples, right? So I told you, you calculate the geometric mean across all your samples. It doesn't matter if it's replicate, if it's condition 1 or condition 2, condition 3. So this has an implication. Do you know what implication is this? When you use all the samples in your data set, right? So the implication is that, let's assume you decide to remove one condition from your analysis next day and focus only, I don't know, you have multiple conditions, right? Wild type, treatment 1, treatment 2, treatment 3, and all the replicas and everything. One day you decide, oh, I want to remove treatment 3 from my analysis. When you remove something from your data set and you do the process again, the scaling factors may change because you are removing one of the replicates, right? So generally what we recommend is always do the analysis with the whole data set. Okay, so whatever you have, the samples you're being collected, do the analysis with the whole data set, even if you have a lot of treatments. So the more samples you use, the more accurate of the normalization process is going to be. If you start removing samples, the size factors may change. I mean, you may need to make that decision later because maybe you have a condition that is weird, extreme, the PCA showing there's a lot of variation in that condition. Then there's other reasons why you want to remove them, right? But the first pass, you should always consider to include everything in the analysis. Don't try to do pairwise comparison where you decide, oh, I'm going to start comparing wild type with treatment 1, and later on I'm going to do wild type with treatment 2, and then wild type with treatment 3, and start splitting your data in that way. DSeq allows you to do the analysis with the whole set at once. Okay, so once you divide the counts by the scaling factors, one of the things that's going to happen, you're going to notice your normalized data is going to have decimal places, right? Probably doesn't make any sense for you in the sense that, okay, now I don't have full counts, but yeah, this is going to happen, right? So you divide by a number, a full count, you're going to get some decimal places in your data. So if at any moment sometimes you get some data from a collaborator and you see that the count metrics have decimal places, it's being normalized. You shouldn't use it with DSeq2 DC2 takes the raw counts, not the normalized counts. Or if you download something from a database, you should download the raw count data and not the normalized count data. Okay, we've seen this. We've gone through the normalization. I'm not going to go through all these details. This is a statistical analysis that I'm not even an expert of. Okay, so what we're doing, right? So here I'm giving an example, right, of what we are trying to estimate. So this is just one gene of our data set. We have a bunch of replicates for what we're going to call, we have some animals that are normal, right? And we have some animals where we sleep deprive them. This is just an example, right? And actually is the example we're going to use today. So basic DSeq2, what it's doing, it calculates the baseline or the interception if it was a linear regression, and then it estimates some sort of change, right? Our fold change. So we're trying to figure out if this difference is actually different from 0, right? If our fold change is significantly different from 0. That's what it's trying to do. And I'm showing this because when we get more complex models, it's for me a list. If I need to deal with a complex model, I like to create my own drawing of how I'm taking the approach, because fold changes gets even more complicated when you're trying to do more complex models. And if we have time, I hope we have time, I'm going to show you a few of those. So I'd like you to have this kind of picture, that DSeq2 calculates the baseline or the intercept in our formula, and then estimates if this difference is significant or not, right? Yes? Questions about this? No? So this is what we're trying to do, estimate the baseline and estimate the coefficient of the condition, so our fold change, and if it's different from 0. And it's quite similar, the approach we do with when we have a continuous variable in the x-axis. We don't have, right? We have a discrete variable in this case, and we estimate the slope. Okay, so I give you an overview of how DSeq2 works, and I think you know the trick here. Oh, sorry, I didn't start the. Oh, sorry, just give me a second. So if you have questions, please ask them now while we wait. So the question about the whole numbers. So we start with the raw counts, which are whole numbers. But when you normalize them, you're going to get some table that looks like with decimal places. I was trying to show that because sometimes you may try to download something from online and you get a table of the counts. And if you see decimal places, it means it's been normalized already. And you don't want to use that with DSeq2. DSeq2 only takes the raw counts. Yep. Yep. Yes.
Sometimes it's in million and sometimes it's in very less number. So there is lot of variations
Yep.
in the expression of the gene.
Yes. Yep.
So that is telling us this much low expression as compared to the.
You shouldn't compare across genes, right? Because of what we said, the length of the gene is also important in this case. If you have a very short gene, you're going to have less read. If you have a long gene, you have a long read. So generally, a gene expression is not designed to do analysis across genes. That's not the idea. It's across samples or conditions. We also have the metadata that we're going to see, but just if you want to double click, you can see the metadata we're going to be using. As the other days, I built a notebook in a way that even if I'm talking and you prefer reading, all the information that I'm saying is in the document. I'm not saying anything new. Probably the document is better expressed because I did it when I was awake, and now I feel a bit jet lagged. So I have the feeling that I'm going to make more mistakes talking than actually if you read these things. But another important thing is, as I said, bioconductor, every package in bioconductor, let's say, needs to follow some rules about documentation. Documentation is great. And they have one thing that I call vignettes, which pretty much tells you how to go step by step through the process of doing analysis. DSIC2 is not different. There's still things that are not always on the documentation, because it's something that you learn by practicing, right? But the documentation definitely should allow you to go through every basic step. And DSIC2 gives you a lot of recommendations. They have an FAQ with questions that I guess that they receive through the years, and they give you examples of how to troubleshoot whatever situation you are in. So there's a link to the vignette of DSIC2 that is very well documented. So you know, guys, what we're doing here with this library function, what we do? Yeah, we call the libraries, we call tools that we may need, right? If we do the basic analysis in R, we are not going to have DSIC2 available. So basically, we're telling, OK, we need DSIC2 for our analysis. Tidyverse, we use it the first day, is the one that allows us to use that percentage, bigger than percentage to do pipes, right? That's the tidyverse way to doing pipes. And it gives us also GG2 plots, where you can make cool plots. We're going to use what we call pretty heat maps, beautiful heat maps. This is just some colors, nothing important. This is a library used by DSIC2. I'll go into details later. This is an open source way to create Excel files. So what I like to do with when we do analysis in DSIC2, instead of exporting tabulated tables, I prefer to create an Excel, so you can open in your computer an Excel file. This is a minor use. You don't have to use it. It's to create. I use it to create interactive 3D plots. And because I don't like Bonnie's way to plot volcano plots, I use what we call enhanced volcanoes. Do you have, by the way, any library that I'm using here, like pretty heat maps, you have other libraries that do the same job. I just pick one that I like, right? But there's a lot of alternatives to everything I'm saying here. This one here is just these options are used to create the window where we plot in Jupyter notebooks. I'm choosing the size, right? Because sometimes the default ones create tiny plots. I want to see the display a little bit bigger. So this is how I say to create a wider or taller display size. Probably I'm going to use this call later on in some spaces because I prefer wider or narrow plots. So just to let you know that that's what it is. Well, I'm telling you what this line does, and that's the only thing this thing does is telling me the time of the day. That's it. So, like the other day or the other days, the only thing we need to do is just execute it. So you can do it. Don't wait for me. You're going to get a bunch of warnings. That's normal. Oh, one thing that you should know about R. Let's see if I found an example here. Yes. Yep. You see this section of conflicts? Basically, it's telling you that there are packages, or when you load a lot of packages, right? So they come with functions, like, I don't know what function we used, like plot function, right? Yep, that they may have the same name in two different packages. So that's what the conflict is, when you have two functions with the same name in two different packages. Yep. So it's telling you there's going to be some conflicts. It's not going to affect what we do, but you should be aware of this issue in R, right? When you load a lot of packages, some functions may have the same name. So actually, the best practice you could do is when you want to call something and you know the package that it comes from, when you call that function, call it with the full name. So the full name would be the name of the package, so Dipler, colon, colon, collapse. So if I want to use collapse from Dipler, call it with the full name instead of calling collapse. Because if you call collapse, you may call it from Dipler or from iRanges, right? And may depend on how the order of things they call your libraries, right? So this is what it's telling you, that Dipler, if you call collapse, you're going to use collapse from Dipler, not from iRanges. That's what this thing is telling you. If you want to use the iRanges one, you're going to have to call it as iRanges, colon, colon, collapse. Because if not, it doesn't know which one to use. So a good practice could be to call your functions with a full name. We don't do it here, but it could be a good practice to do. Let's talk a little bit about the data we're using. The data comes from this paper, and I hope I remember the details. So what they did, they created a knockout, right? The Shank3 knockout, Bonnie, I think that's what they did. And then they put the animals to some sleep deprivation. And they did the comparison, and they come out with some conclusion, right? Related to circadian rhythms, I guess. We don't care much about the goal, so we're going to create our own goals in this analysis. But just to let you know, if you want to know details, the data comes from this paper. So it's the original data they use in this paper. And you're going to notice we're going to come out with a different conclusion. Not the one in the paper. I never went through the paper to know exactly how they did the analysis. I don't remember the details. But it's not the same results we're going to get. So I guess that happens in science. We are not following the same analysis steps they did. But I'm wondering what they did. I don't know. One day I need to redo the analysis in the way they did it. I never done it. We never have the time, right? But I don't think they use DSeq2. But if you have time, if you click here, it's gonna you're gonna go to the paper, so we can check exactly how they did it. So another thing I did, so I created the row counts myself. So these are analyzed pretty much in the same way you did yesterday, which it doesn't mean is the way they did it. So this is how we're going to be reading our count metrics. So you use already this, right? Read CSV. And we're telling that they use the first column for row names. So these are our genes. And everything else is going to be transformed to a matrix. That's not important in this case, because DSeq2 also would do the transformation for you. But instead of having a data frame, we have a different structure, which is a data matrix. Oh. And why we do this, right? Why we're doing this? We're getting the first rows. By why we do it? We want to be sure we are loading the correct data, right? Always visualize what you do is always good. Be sure that you created the right object.
Why that is going off?
I guess it goes to sleep. So if I want to, oh, by the way, it's too small or not in the back? Should I increase this? Okay. What about now? More, less? More? Okay. Is this the same? Whatever it was here before is the same as this or not? Yes. I'm just printing the count data and had only taken the first two. This is a pipe, right? Where we said, actually I should do these all the time, but I keep forgetting. So in that way we get used to this concept of deep learning. I recommend it. It's a really cool tool, and it makes reading what you're doing, I think it makes reading your code easier, right? So I get in the table, give it to head to just print the first 6 lines.
So what others, like when you are drawing the head, you can also run like head, parenthesis, then count data, same thing, and then, like what you guys did yesterday. So both is actually same.
Can you please expand it a little more?
Uh,
Please, slightly more. Zoom.
Slightly more zoom.
More zoom. I'm going to run out of screen space. Can I?
Maybe one more, right?
Yep. So we got the table. So the first issue is that, right, we have a really weird names for samples that doesn't tell us much. So we're going to have to deal with that later. But yeah, it looks like we uploaded the table. Another thing we mentioned yesterday, the gene names, right? They have double annotation here. So we have the ensemble ID for the gene and the official gene symbol. It's mouse. Yeah. Sorry, I should have told you this. We didn't sleep-deprived humans in this paper. We sleep-deprived mice. I'm sleep-deprived, but the mice also were sleep-deprived. You keep going. I'm not going to read this. So I think for you guys, the best thing you can do, it's easier for you probably, is if you create your metadata in Excel and read the file, probably, or a tabulated table. Generally, it's better if you create, don't save it as an Excel file, save it as a tabulated table. It's easy to read. That's probably my recommendation. And you create your metadata. If you need to create your metadata, right, you create that table on Excel, for example, and then you import it to R. But if you want, in R, you can create data frames, right? We've seen this before, I think probably the first day. This is a vector where I'm giving the sample names. I'm creating another column telling it if it's what type of the donor of that sample. I'm creating also the treatment, if it's been treated or not. I'm assuming it's what I did here. And then I'm creating a data frame with these 2 columns and adding the sample names as the row names for that metadata. If you run this is a way to create a table in Excel, right? In Excel, in R. Right, so all that code creates this table. I have the feeling for you guys, if you don't want to deal with coding, you can just create a table in Excel and then import it to R. Oh, did we import the metadata at some point? Oh yeah, what I was trying to say earlier, right? So these sample names doesn't give much information about what they are because they were downloaded from GEO. These are the sample IDs. This is the metadata, the file. Any questions? I'm going too fast or not? Sorry, I probably should stop in every step and explain what we do. You know that read CSV, right? I'm just reading the table.
Metadata, or should we can name?
Oh yeah. So the objects that I'm creating, you can name them as you wish, right? I'm just giving names of the terminology because I think it's easier if we talk about metadata, call it metadata. But actually, DSIG2 call it call data, column data. They call it call data. But I think metadata, for me, I understand it better, so I call it metadata. But you can call it whatever you want. And the only thing we're doing here is just reading this is the metadata, the file we saw earlier. Again, the same thing. The row names are the first column. And every string here is treated as a factor. So It is not treated as just text. It indicates something. It's a factor of your discrete variable, right?
So it has written true in the command, strings as factor true, right?
So you've got two conditions here. One is that normal and sleep deprived, and the genetic background of the mice is also different. One is wild type, and what is the SHANK3?
It's a knockout of the gene JANK3. Yeah, so SHANK3.
It's required for synapses or something like that, for neural synapses.
I don't know what SHANK3 is required. I'm not sure.
It is required in synapse formation.
Oh, okay. Oh, okay. So SHANK3 is the name of the gene that was knocked out. Sorry if I haven't been explicit about it. So the metadata.
String as factors true. Why do we put true?
We'll talk about it. Just execute it. Don't worry about it. Google it. Or if you don't want for me to explain it, you can Google it. But don't worry about it. It's not important now. This is the metadata. So we have the genotype. We have the condition, being sleep deprived or not. For the purpose today, or the first part today, we are not going to care about the genotype for now. To make our analysis simpler, we're going to care about if it's being sleep deprived or not. So we're going to focus our formula, right, is going to be that we're going to assume that differentials, the gene differential or the differential expression we observe in our data is going to be caused by the condition, being sleep deprived or not. That's going to be the assumption today, or for the first part. You have the explanation next. Another important thing that it's a good practice, again, you don't have to, but I think if not, you may encounter problems later, is to level or relevel your factors. So our factors are really simple. We have only been, so remember we said the condition are considered factors. Our factor is being normal, normal or sleep deprived, right? So whatever we use as a base, right? So I think DC2 takes it as alphabetical order. So I don't know how we call it, normal and sleep deprived, I think are the two conditions. So normal is going to be considered our baseline. But let's assume you have a lot of treatment 1, 2, 3, 4 to 5 or different names. So you can be really explicit about what is the first level in our factor. So let's be explicit just in case, and we can practice it. So you remember what this means? So metadata is our data frame, and we are calling the column genotype, right? And we are releveling the genotype column, indicating that wild type is going to be our base level. Now, you're going to have to relevel the other two. What is going to be our base level? Well, yeah, yeah. So double check whatever the metadata says, right? So if you type normal with uppercase, it's not going to know what you're talking about, right? It needs to match whatever is in your metadata, which I don't even know if I type it correctly. Again, everything I'm explaining is explained before or after the code, right? I mean, you can even have the levels in a specific order, like low, medium, high if you want to, and so on. So let's do one thing before this happens again. Try to relevel, and I'm going to plug in the computer so it doesn't go to sleep. Thank you. is normal. As a reminder, remember the trick using autocomplete and tab. So if I start typing metadata and I press tab, it should give me all the options for keywords that start with meta, for example, metadata. So you don't have to type every time the whole thing. It's normal. Are you copying me? Don't copy me. I may do something wrong. This condition is normal, yes. And wild type is the other one, yep. Is wild type? No. Is wild type normal underscore normal. Any issues relabeling? You are not going to get any information if what you did is correct or not. So again, if you are new to coding in R, it's always good to check what you're doing. So you could, I think if you call level or levels in plural, I would. Oh, no, it's levels. If you want, you can check the levels in your columns by just typing levels and the column that you want to check, and it should give you whatever is whatever you decided was the first level, right? So this is a situation where maybe, right, the column genome condition, which is the combination of the condition and the genotype, where you have multiple levels. This is an example where you may want to resort them in a specific way for whatever reason. You could, right, because you have multiple ones. But in the case of the genotype, you only have wild type and another one. So if you know that DSIG2 takes them alphabetically, you may skip this. I still think it's a good practice to stop and think, what is the baseline? What is your baseline? What is your reference, right? So it's not bad to use explicitly and tell what you're doing.
Commands at the same time. Animal use will be right type and the condition used will be normal. That is my reference base level.
So sorry, start again. I'm slow today.
I'm taking the two parameters at the same time in account by giving this command.
Which command? The level one?
Genotype as well as the condition.
Oh, you mean the third column. The third column, yeah, I should have explained better. Yeah, the third column in the metadata. Yeah, I should have explained better. Sorry, yeah, I should have stopped and explained this. My bad. So first column is clear, genotype. Second is condition. What I did here is to combine these two in a single factor. That could be another way you could do an analysis, right? We can do the analysis considering genotype alone, condition alone, combine them in a way that we have 4 different states and mix them in one. That could be a way to do the analysis. Or we can use complex formulas and use the genotype and condition in our model separately but in the same analysis. So I guess my recommendation is don't do that initially, but there are ways, I mean, you could do it. And actually it works well too, right? What I tend to do is keeping everything separated and do an analysis like that. But again, this is normal, it's fine. I mean, you're not going to get wrong doing the analysis.
Expand your table, making multiple columns and then pick.
Well, you don't have it. Yes, you don't have to do these for every analysis, right? But I'm saying, let's imagine you want to do pairwise comparisons. That's what you're going to do, right? And you want to compare, for whatever reason, wild type normal, versus versus the knockout normal 2, then it's easier to create a column with those data and then compare them pairwise. But if you want to know, let's phrase it differently. If you want to know the coefficient factors of every variable in your metadata, you need to have them separately, right? If not, you're just mixing everything here.
No, but the genes, what we are referring, what we are finally analyzing, that will be according to this one condition, right? This is just to name the our group.
Yeah, so here you're creating the grouping that you want to analyze with this table.
Yes, and the genes are per group. Whatever genes we are analyzing.
You're creating the groups here.
Yes.
This is the definition of the groups.
Yes.
Yeah.
So
So
every comparison in our data set or in this data set, it's between the groups.
yes.
And the genes vary between the groups.
Yes, however you define the groups, which it could be just taking one column into account, another one, combine them both together. But the main issue of combination is that you're not going to know the exact effect of wild type and normal separate. You're just mixing. Then you need to make the internal effort to interpret that data of what it means when you do the pairwise comparison. But DSIC2 have the power of telling, if you give me these factors separately, I can estimate the coefficient for genotype and the coefficient for condition separately. So that's why I guess my general recommendation, if you have this type of situations, first try to do separately, and then if you think that it's better to combine for whatever reason, and there's reasons for that, you could do it. So no problems relabeling, everything is good. So next part. Still, we haven't touched DSIC yet. So we're just doing checks that everything looks good, our metadata looks good. So another step is to see that metadata and count data are compatible, right? So any thoughts about what is happening here? The first line. All is going to just return true or false.
So rows have data, the columns. We are defining our columns and our rows.
We are not defining them. So this is going to return the row names of metadata. This is going to return the column names in our call data, right? The row names in the metadata, what they are? No, metadata, the samples. Yep. They are the samples. The call names in our count data, what they are? No, the samples, true, they are the samples.
Sample 1, sample 2, sample 3.
Yeah, so these are samples, these are samples. So basically we're saying that the metadata table that we have, the row names or the samples names are present in our count data. So we are checking there are not misspellings, nothing weird. So basically if all of them are present, all returns true. That's what we're doing here. Okay, next one. Well, I'm already saying what it's doing here a little bit. We've seen this before, I think, at some point. So this is our count data. The way to access the count data is by row or column, right? So we are giving our samples to the count data, saying we are subsetting our count data to only use whatever is in the metadata. That's what we're doing, and store it then in our count data. So if our row counts have more samples than we have information for, basically we're excluding them. So we want that all samples in the metadata are represented in the count data and the other way around. We don't want extra information because DSIC is going to tell you, oh, you're giving me extra samples, what I do with them. So we are subsetting our count data by the column names that are present in our metadata. That's what it's doing, right? And storing that in count data. In our case, it's not important because everything matches. So there's nothing happening here. But it could be a case that you have a count data that has a lot of samples and you only care about a fraction, or that's the one that you have information in your metadata, so you're subsetting. So the square brackets, what they're doing is the first value in the before the comma is saying take all the rows, because I'm not subsetting. The second is take the columns that are in the metadata. That's basically pretty much what we're doing. What about this one? So it's similar to the one on the top, but it has the identical. Any thoughts? I mean, there's no wrong answer. We haven't seen these before, so they need to be in the same order. So if you read the text, I mentioned that they need to be in the same order. So this is checking that it's in the same order. This line is useless for one reason. Because when you subset, actually uses the order you give it. So it's taking the order that is in the metadata and sorted the count data based on that order. So this is just going to return true no matter what, because we run the second line. So if you run, it tells you, yep, so it's true, everything is present, we're good. It's true, everything is in the same order. Just a sanity check. And if again, everything I'm saying, and now I'm realizing everything is here, so I'm not, if you notice that I'm explaining something right, give it a look at the text, because it's explained. I'm probably better job than I'm doing. Not much. A few seconds. Less than a minute, I'm assuming. We talk about this, right? So I said we're going to take the simple example that our expression data is based only in condition, beta has been a sleep deprived or not. That's our expectation. So these are going to be our formula. I always like to put examples of how you phrase your experiment. I think that helps when we deal with investigators, right? I'm giving, let's say, different levels of expression. So with that formula up there, right, what we're saying is that the gene expression levels depends on the experimental condition, being sleep deprived or not. If you want to statistical phrasing, the models assume that the variation in expressions can be explained by the condition variable. That would be, and if you go the equation style, right, that would be how internally R read it, right? It says the expression of each gene is its model and as a function of the experimental condition. I don't know. I have the feeling that sometimes expressing what the model means helps a lot. And we're going to do an exercise later about it, especially if we take into account more complex models. So I always try to, when we collaborate, try to make the investigator to express what's the goal, right? Our goal is we are assuming that our condition changes the expression levels. I know it's really simple, it doesn't make much sense now, but if you have a complex metadata table with a lot of factors, it helps to phrase it a little bit, or make the exercise to phrase what is your goal so you would understand how to build the model. Okay, what we finally expected to run, right? So you don't see it here, but you can see the let's do the proper. In R, for example, you can break lines. If you break them in functions after a comma, and not going to be a problem reading it. We don't need that / like we use in Bash. So it's clear what we are providing, right? We don't need to go through this. So the first thing is just telling, read the data matrix we created object DDS, and now this function here does everything I mentioned, normalizing and doing the statistical analysis. It does everything. So And I hope it doesn't crash. AWS is giving me trouble today starting it. I hope we don't have any issues. Did any one of you finish the whole thing? And it's okay, so some people already finished. Don't worry about the warnings. If you type DDS, which is the object that we created, you're going to get some information like this. So it's not going to print the whole object out, but you could access all the information inside. And we're going to see how we're going to access that information. But in here, you have everything, like the normalized counts, analysis, et cetera, et cetera. So we already talked about the size factors. That's the size factors, that complex steps that we took on the presentation that is used for normalization. You could check them, right? So every sample is going to have a size factor. So the way to access, the way to access is with this called size factors, DDS, and it will print out the size factor for every sample. The as data frame is just to make it nicer to look at it. That's it. There's nothing special about it. And again, I probably could do this to make it, to be consistent in the way we should do it, right? So it's going to be hard to show on the projector the values. I think I'm asking a question, right, up here. Can you guess which one is the one that has been most sequenced or has higher depth of sequence, and which one is the lowest sequenced? I don't see the whole table. You're going to look at your own table because I don't even know. Remember, the size factor is what DSIC2 is dividing your counts with. Any takes on what sample? You don't have to give me the name of the sample. You can use the size factor to figure out which one it is. Which one is the most sequenced? We are dividing our row counts with the size factors. Which one do you think is the one most sequenced? Remember, normalization takes into account two things, which is the sequencing depth and the composition, based on the steps that I mentioned in the slides. The one with the highest is going to be with a higher number, right? That's, you already can tell which one is the one that has the highest depth. I'm assuming it's this one. All the numbers look like. Yep. And this is the one with the lowest value, right? Probably. Yep. So that is the other way around. This is the one with the lowest sequencing.
So like pretty much the size factor basically tries to make the amount of rows we have uniform, right?
Yep.
Like it's kind of basically
To be able to compare them, right?
Yeah, it's accounting for like that some samples have more rows because we like got more data.
They have more counts, no more. All of them should have the same rows in theory in our table. So the ones that have more counts, yes, that's what I'm trying to do. In addition to the problem of the composition that I mentioned, right? Ideally, right, if you do a good experiment, you want everything around 1, because then you don't have to force the normalization process to change your numbers, right? That would be the ideal. So can you make decisions based on the size factors? Normally, we don't. You could, in the sense that you could keep track of saying, oh, maybe this one has a very low number. It could be an outlier. It could be that we didn't sequence enough, right? And when you do PCA, if you see that sample, you know, in one of the extremes of your PCA, you say, oh, maybe that sample needed more sequencing. The size factor are pushing too much the data, and it's creating artificial signal, right? Normally, you don't have to make those decisions, but you could check them, right? So what we're doing here, R can be easy to read sometimes, and I think this is the case.
So it is based on the count data.
We're taking the count data, yes. We're not taking the DDS object here. Yep. It's just called sums, right? The sum of the column and reporting. So if you want to double check what we said, this is the sequence. This is the more or less the sequencing depth. That's the counts that we got, right? So you can check if we were right or not. I think this was the sample with the highest depth, which is 7 million on counts, and around here, yeah. That must be the, no, one of these is the lowest, I'm assuming, around here. Yep.
So these are the count of genes.
Those are the counts of the genes, the sum of every column. That's what it does. It doesn't do anything. Again, if you don't work for me, you can keep reading. Everything I'm saying is on the Jupyter notebook.
Count of genes. Sum of count of genes. Right.
Call sums just returns the sum of every column. It gives you value for every column. Every column in your count matrix is a sample. Again, I'm insisting on the formula, but we already been through this. This is another way to represent it, right? I like a lot the way to use the arrow system to have it in mind, meaning, so this is our formula internally how R uses the linear models, which is calculating the intercept. If we do linear regression, you can call it intercept of the baseline. So it's estimating what is the value of this. So from 0 to my average for that condition, and then estimating from the baseline to the average of my treatment. And these are what we call the coefficients, right? Or in our case, this is going to be our fold change. What is the fold change between the baseline, control, and treatment? But I like to use the arrows because, again, if you, even in a simple example like this, which you don't need to do this because you already have the mental image of I'm comparing two things and I want to know the fold changes, having even a simple example, what happens internally is going to help you to understand more complex examples. Or not because you don't care, because you always deal with simple examples, you don't need to do these things. Okay, so how we access we could, again, everything that DSIC2 needs to do is already been done. The only thing we are doing is exploring the object. We could access the coefficients, which coefficients in this case are the fold changes. So if you do this, you're already accessing the coefficients we are calculating in our formula. This is the baseline. What is the baseline? The average expression in our normal. So this is our baseline. And this is our fold change. And yeah, in the way that it's built, yeah. So it's readable because we're giving it readable columns. If we give him something weird in our metadata, it's going to be harder to read. But basically, DC2 creates this. So I'm taking condition, the column condition, and I'm comparing this versus this because this is our baseline, and this is our fold change. You probably are not going to do is all the steps we're taking, you're probably not.
Why is it showing only for the 6?
Oh, because I'm using head. So head, so again, let me do it. Let's be consistent. I don't know why I wasn't consistent this day when I type all this, but I want people to get used to this. So if I only give it the first part, coefficients DDS, would print everything. But I'm giving everything to head, and head only prints 6 lines.
So here we are getting the average of baseline. The value is 10 suppose.
Yeah, so
And the fold change between two conditions. But we are not getting value of the condition. Like it's 10 for the baseline.
So,
value.
sorry, what do you mean? I'm not following the question.
So here, this 10, what does it represent? It
So
represents
for you, so the simplest explanation of what is 10 is the average of your baseline, normal. That's the average expression of normal. That would be the phrasing
yes,
for us, for normal people.
what about the average of the conditions?
You can calculate it later, but this is not what it's doing. This is giving you the coefficients of your formula. Your formula is baseline plus condition. So baseline plus your coefficient. So this is the beta. Did it die? No.
For one gene, it was showing NA. Why was that?
You probably don't have data. So there's no counts for normal, there's zero, or there's another reason. It could be, it's, so every step we're taking, you don't have to do it in your analysis. We're just exploring the data. You're barely ever going to run coefficients. You don't care, right? Generally, we're going now through all these steps is just to explore and understand what is happening a little bit in the background. But using the coefficients part of it, you barely ever, I don't do it. The coefficients are on your final table, are your fold changes anyway, right? You want to know the p-values, the fold changes, you don't have to do all this. It died for everybody, but it's me. Okay. But again, if you have a complex model, right, this table is going to be much bigger. You can ask if an example we had yesterday, right? It was genotype, sex, and condition. This table will be much bigger. You may want, you generally wouldn't do anything. Oh, okay, it's going back. Oh, sorry, in the middle. Let me, wait. So any questions? So the only thing we do with the coefficient is to calculate this and this, the beta 0 and beta 1. Well, we're not calculating, sorry, we're accessing it. So, and as we said, this value is just the average of the expression in controls. This value is our fold change from beta 0 to beta 1. So where we ended up in here, right? That's what we did. Visualizing the coefficients. Again, this is not a step that you're going to do in your analysis normally. You're just, we're just exploring the data, right? So you get familiar how, if you need to access, you could. Hey, I'm using the mic, right? Oh, okay. Should I get it closer? What about now? Better? No? Good? Should I speak louder? Okay, I speak louder. I blame you if I'm losing my voice already, and it's only Wednesday. Yep. So again, if I'm going too slow and you want to read, you can keep going. You don't have to wait. But I'll go step by step. So this is another thing, another complex thing that you don't need to know, especially if you're dealing with the most basic analysis, which is just comparing within a condition to treatments. But internally, the linear models, you use this matrix that you see here, that it's going to be hard to show in a single on the screen. I'm going to make the text. Sorry for the people in the back. Yeah. So internally, this is another way to look at the formula, right? So this is how your grouping is working. So actually, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 samples, which are our normal samples. And basically, internally, R is reading this matrix and saying, okay, how I calculate the intercept? What samples are the ones that I'm using for the intercepts? The ones that have a 1 here, these are the ones that are normal. And you are not using them. We have a 0. You multiply by 0 to calculate the coefficient. Now, okay, how do I estimate where beta 1 ends? So you scroll down. Okay, these samples, they use 1 arrow plus a second arrow. These are the ones that belong to our condition. Now, again, this probably you're not going to use it that much. I'm showing it. Again, this is a simple thing. You only have one condition, normal plus treated. You don't need to reach these details in your analysis. But if we have a really complex model, and if we have time, I'll show you. This helps me, at least helps me, how to draw those arrows and understand how many coefficients the ESIG2 is going to calculate. So if you want to know how internally it's calculated or access the model matrix, it's through this function call. Okay, ESIG2 recommends some pre-filtering of the data. So we said 60,000, right? Ensemble, something like that, between 60,000 and 70,000 genes annotated. So if you do a volcano plot like Bonnie did, you're going to have 60,000 dots, and it gets really crowded. So one thing you could do, you could remove from your data genes that are very low counts, which ESIG2 is not going to use anyway because they're really low counts. So for your visualizations, you want to remove dots from your data because there's going to be a lot of genes with 0 counts and things like that. You could remove them. That's one thing you could do. So this is one of the ways you could do it. So basically, it counts how many samples per row, right? So your samples are columns. How many samples they have more than 10 reads. If you have, in this case, I'll limit it to 10. If you have at least 10 samples with more than 10 reads, you keep that gene. If not, you throw it away. I don't care, because there's not enough information for me to do the analysis anyway, so I'm not going to use it, and you throw it away. So this could be a way to do it. I'll show you because this is one of the recommendations they do. I don't do it. Why? If we do this at the very end in your analysis, when you get your table, those genes are not going to be represented, which is fine. But sometimes I get questions, oh, what is SHANK3? I'm making this up, right? SHANK3 is the gene we're dealing with. I don't see it in my table. Well, my answer is I filtered it out, right? If I don't filter it now, it's going to show up with zeros. But at least the investigator knows that the gene doesn't have any information, so I know why it's not present. But it's up to you. We can do it or not. It's fine if we do it. Anyway, we are not searching for a specific gene, so we can do it. So we went from, in this case, we actually have 20,886 genes. We went down to 15,248. Actually, internally, and I think I give some details about it if I remember correctly at the very end, internally, DSIC2 has a way to remove unimportant genes because they have low counts. So you don't have to make a decision, arbitrary decision of saying, I'm removing these genes because they have low counts based on an arbitrary number. DSIC2 uses different approaches, and you can even select what approach you want to use to remove genes that are not represented enough. So internally, DSIC2 deals with it. Okay, quality control. This is another recommendation, which is to reduce the effect of the variability when you have low counts or high counts, right? So as you can imagine, if you have low counts, the variability is going to be higher across all your samples, replicates, and all that. So there is one transformation that we can use. There are actually those transformations, right? So this is called the variant stabilization or the R-log transformations. This is quite common. You also use these types of transformations in other fields, in single cell sequencing and all those. So these transformations are only used for visualizations, basically. When you do your heat maps or things like that, it's good if you transform the data first. And what it means is now we're going to have our original data and our transformed data. So we're going to have two counts. So we have the normalized counts that is being used for analysis, which the analysis is already done. And we're going to have our counts being transformed with one of these two systems, the variance stabilization or the R-log. Which one I'm using in this case? I think I'm using the R-log transformation, yes. And you can use the BST. So if you want, you can comment out these two lines and use the other one and see what happens. You can see if we notice a difference. Again, this is only for visualizations. I think the SIG2 recommends VST when you're dealing with a lot of samples. We are not. So any of the two should work. And then we're still doing the exploration data analysis, right? We are not dealing with p-values or anything like that. Everything is already being calculated. We're just exporting the data. So like in PCA, so the next step is going to be our principal component analysis. Instead of using the original data, the normalized counts, we're going to use the transform counts. And again, the main goal is to remove this variability so the variability on lower counts is going to be much higher, and the variability in higher counts can be also higher. So we're trying to reduce the effect of those and focus on the main genes that are in the middle, which are probably the better, best. It's working for you guys? Still thinking? Well, another way to know if it's busy or not is if you see a full dot, I think it's thinking. And also you have it here, like it's working. Let's give it a minute. Yep, it seems, I mean, we're keeping the CPU busy, so I'm assuming it's doing the calculations. So meanwhile, should we review the principal component analysis, what it means? That's the idea of dimensional reduction. So we have 15,000 dimensions, so this is the number of genes, and we are trying to represent the same information in a plot that only has two axes, right? PC1 and PC2, for example. or 3-axis, because I'll show you how you can do a 3-dimensional thing. Sorry, today AWS only allowed me to choose a relatively small computer for whatever reason. So that might be the reason that it may take a bit longer. Questions? I feel uncomfortable being in silence. Can you? Well, first, this needs to finish. Did it finish?
Okay, it take time.
It may take time. Again, if you see the asterisk here, it means it's working. If there's nothing wrong with the website. So until you don't get a number, we should get a number that is after whatever number we had before. Then the PCA. I mean, you can execute. Actually, you could execute the whole thing, and it should work step by step. Except that sometimes you need your input. So. But once you fill all the gaps in the whole notebook, you could execute the whole notebook at once. And it's going to go step by step. For whatever, I don't remember being taking that long, but it could be. If for somebody finishes, please tell us so we know that it's working fine. And oh, it finished for you? Oh, okay, then we only need to wait. Okay, that's easy. So for plotting PCAs, I'll show you 2 ways. This is the internal function from DSIC2 to create a PCA. And we're going to use a manual one or the R1 way. Basically, because I thought it's easier for me to access. You remember the script plots to know how many. So the percentage of variance of explained by each PC, for me, was easy to access through using the standard way of R. But you can, if you only care about a plot, simple plot, you can use the one provided by DSIC2. Did we get results for anybody else? Only for the smart ones over there? Are we saturating AWS? I think AWS is smoking now. Yeah, it's 100% everything, okay? Yeah, we're reaching the limit of the... So if you keep reading, please, exactly, read the next section. So when we reach it, it's going to be easier to understand. The one about principal component analysis. And have your questions ready if you have any. So somebody yesterday was asking me, oh, what if we reach the limit of the CPUs in the system? A lot of people using it. I think we have an answer now. So if you're curious, we're currently using 32 CPUs. So we have 40 people requesting a job. So definitely the computer is on its limit. questions? Did it finish for anyone else? How did you do it? You started earlier, I guess.
No, we ate at the same time. It was
Listen.
just luck, I guess.
Okay. Is any of you a stand-up comedian and wants to come here and explain some jokes while we wait? Or anyone who wants to hear a story from Bonnie, the long ones? You're too quiet today.
I'm finishing some of my work.
Oh, that's what it is. That's why you're quiet. And you are not interrupting and saying something that I already said in the previous slide?
He's banking.
Is,
No, actually, Francis.
yep.
So we have to actually ask them a question. Like, so you all are doing this transformation like VST and RLog transformation. Why you need to do transformation? Why you need to transform data?
I guess it's more genetic than what I said before because you need to.
I don't know whether they really appreciated that what you were talking about dispersion. That is why I thought that we have
I
a
think that's an insult, but yeah, go ahead.
no, like, so what do you think? Why you do?
The compressing by log transformers.
Yeah, why you need to do that? Why?
Sorry, this is not what I want to open.
So many large numbers will be flashing.
No, I think he said that we are converting the outliers.
Yeah, but I think he wants a more generic answer than the specific. He doesn't care about if it's RLog, BST. You're talking about general transformation of data?
So it doesn't mean that. So people do all kind of transformation. They have statistics and things like that. But what we are really, why we need to do that? I mean, if you have a data, we captured it. We just put everything there without making any change. But why we need to transform that?
I mean, the large big numbers count. So we are giving a smaller number by log transforming, compressing, and we will bring in more uniformity.
More clear image visualization.
Or better comparison.
So actually what happens that in most of the data sets, like when you have the gene expression level, how much actually gene expression, lowly expressed gene have higher dispersion. And then, so what happens really, you want to actually control the dispersion in your data. So you are, by transforming, you are actually pushing that low expressed gene further to the line and then log for that. So essentially what you're doing in all this, whatever
Yeah,
you're
the
doing,
battery is
VST,
gone though.
log or any other, let's say, other kind of normalization, which is actually the linear model.
I kill it.
So all this kind
He
of
deserves it.
normalization, the idea is that can you control the dispersion in your data in such a way that the gene expression itself should not actually impact the level of gene
It's
expression
hot
should
again,
not
right?
impact your differential gene expression or significance form. That is actually the idea there. Controlling the dispersion in a way that the lowly expressed gene or highly expressed gene should not influence your significance calculation.
In this case, the transformation is only for visualization. The transformed data is not used for analysis. The analysis is already done. We already done all the analysis. Sorry, yeah, I dropped it and I lost the battery. It's below the thing. Yeah.
No, refill that in the bag.
How long it took only 1 minute or two for you, right? So one thing I'm gonna do you mind to, I don't know, should we try to half of the class stop it and the other half run it and see what happens?
Alex, what we can do, this is like 11:15. Francis,
Yep.
let them run, and I think I will actually let this keep running. I will actually start the NGS class, and that will give them some time to finish all of it.
Well, we hope so. And it's not stuck for whatever reason.
Let's see. Okay, so let's do that way. I have sent you a presentation.
Oh, it works even here. Yeah, that's cool. You can scroll up and down. Well, let's get ready for Bonnie then.
Come, come. You have to start. Okay. Yeah, just put those slides somewhere, anywhere, and then I will rearrange it. Anywhere, anywhere, just put it. I will, or will. Okay. Yeah, just before clinical application. Yeah. Yeah. Okay. Yeah.
So while your code is running, just a few things. So when we think of transcriptomics, there are three kind of layers to it. Bulk transcriptomics, single cell, and spatial. And the widely publicized analogy of a bulk is like a smoothie. So what you get is that you don't, I mean, in a smoothie you put everything. You put strawberries or whatever fruit of your choice, bananas, and you don't know what taste is coming from what. So the analogy of a bulk transcriptomics is to a smoothie, whereas in a single cell, it's like a fruit salad maybe, where you know exactly, if you want to taste, you know how does a kiwi taste, how does a lemon taste, or a berries or mangoes or whatever. And in spatial, you also have information not only about each single cell, but also about the spatial orientation of that cell. And it's widely used in pathology actually to know about the single cell spatial transcriptomics. So in terms of immunology, when you look at the different uh, ways that you can employ single cell genomics or transcriptomics, you can look at the different antigen receptors, like you can do a T cell receptor or B cell repertoire analysis. You can look at cell dynamics, you can look at the interaction between cells. You can look at cell-cell communication. And if you're doing single cell spatial transcriptomics, you can also look at the tissue architecture and the cell-cell communication. And in that way, you can dissect the heterogeneity within a sample which would appear to be homogeneous. And the essence of single cell transcriptomics is similar. So you first do a sequencing, you get the reads, you align the reads, you do a quantification as you do in bulk, and then you do QC steps. One of the QC steps is to look at the reads coming from the mitochondrial genes, because you don't want that. I mean, there should. So one of the QC steps is that you should not have too much of a mitochondrial gene expression in your sample, because it indicates that a poor viability of your sample. So what is the threshold that you generally use for this, like for mitochondrial, or do you filter out on that?
Yeah, we usually filter out. I think Francis will be the one doing it.
Well, it depends on the sample.
Okay.
It could be 10%, could be 20%.
Or you can even relax that? I mean, even 20% would be good enough?
I mean, depending on your situation,
Okay.
you need to deal with, you're looking for a population that is very low in your sample, and you need to keep as many as you can. You probably need to relax the frequency of filtering.
Then you do a normalization, as Francis talked about, and then you can do clustering. So you can see that different cells, based on their gene expression, do they form distinct clusters. But apart from gene clusters, I mean, clustering according to gene expression, you can also look at trajectories or linear trajectories where, or based on the gene expression, which cells are rising from which progenitor cells and all that. So you can do a trajectory analysis based on your single cell data. You can also look at regulatory networks when you have single cell data. So these are some of the applications from the single cell transcriptomics. You can look at clusters, like are there any distinct clusters based on gene expression. You can look at trajectories, what cells are derived from which cells, are they related to each other in some way. You can look at regulatory networks, and you can also look at cell-cell communications. And in terms of spatial transcriptomics, so this is from from a kidney sample. So in a single transcriptomic map, you can have a spatial reconstruction using either the landmark genes, or you can do it de novo. And then you can also look at cell communication networks. So in this case, you have got like pelvic epithelial cells from the pelvis of the kidney, neutrophils and macrophages, and you can look at the different cytokines or chemokines which are used to communicate between these cells. And the lower figure, you can see the nephron, which is divided into three parts: the cortex, the medulla, and the pelvis. And different fluorophores are used here: phalloidin, Lis-M, and a red fluorophore for CD11B. And so, based on this, you can look at cell networks. You can do a spatial reconstruction. This was actually a transgenic mice which had a susceptibility to pathogenic E. coli. And so the Lis-M was that mice, and the merge shows that where the CD11B and the Lis was, that shows off as yellow, and the phalloidin in the cortex and the medulla is showing as white. So in spatial transcriptomics, there are different methods to do it, different platforms to do it. So, but that you can do it. The single cell spatial transcriptomics can also be captured at a single cell resolution. So I think that's what I wanted to talk about. So you will be.
Yeah, I will be talking more about the spatial transcriptomics as we finish this talk class. So this will be the last one for this year for the NGS technologies. So we were actually yesterday we were discussing about the applications of NGS, and one of the very recent application is proteogenomics. That transition, slide transition. Oh, okay, thank you. Okay, so the slides got a little shabby when we are actually transferring it. Okay, anyway, so one of the way to do this, there are two different approaches how you will do the proteogenomics. Okay, so proteogenomics stands for studying protein or quantifying protein or both identification and quantification of protein using genomics. So till this time, most of the protein identifications are done through mass specs or low-res ELISA kind of a technique. But there were no way to do like thousands of protein assays on hundreds of samples together.
and the proteogenomics is actually opening that window. And there are two different ways to do this. One is called as SomaLogic, or which is a company called SomaLogic, which is recently purchased by Illumina. And sorry for being this bad slide. Actually, it was not like this when I made. So in case of SomaLogic, what they are doing, they are using something called as aptamers. So aptamers are DNA molecule. Okay, they are actually nucleic acid, and those nucleic acid can recognize proteins. So essentially, what is happening that the DNA or nucleic acid makes a tertiary structure, and that gives you a binding pockets. And the base, you can change the sequence, and you can actually change that binding pocket where protein can specifically interact. So that is a very specific. So interaction of aptamers with the protein is very specific. That means you design aptamers unique for each protein, and that will give you, it's kind of an antibody protein interaction. But the good thing about aptamer is that antibodies are still sloppy, but aptamers are very, very specific. But then the problem with aptamers is that it needs a lot of designing and validation. But once you have an aptamer, then its specificity is amazing. So it's not as sloppy as. So this is an aptamer. They have some modified bases and normal base. And in this reaction, what you do, this aptamers, it has a biotin molecule here, a linker, and I will tell you why all of these things are, and a fluorophore. So NGS based method, you don't need that fluorophore. They are there for microarray based. So this platform first came for microarray. Now this is also available for NGS where this fluorophore is not used. Now what you do, you immobilize these aptamers on a streptavidin bead. So these are biotinylated aptamers. So you immobilize them on a streptavidin beads. So once you, and then you add your protein, mixture of protein. So if it is urine or plasma, whatever it is, you don't have to isolate protein. You can actually add the real material like plasma or urine or cell lysate. I mean, you can't do cell lysate with SomaLogic, but with the POLINK you can do that. So you add them. So protein will go and bind to its specific aptamers. And those are immobilized on a streptavidin beads. Now you pull everything out. You wash anything that is not bound, any protein that is not bound, you wash them off. And now you have a very specific interacting aptamer interacting with protein with all this, linkers and all those kind of stuff.
Why didn't it work for a protein lysate? If you took the cell lysate,
yeah,
they extract the protein. It works for that.
Yeah, it will, but the whole idea is not that. The problem here is why we don't do that, because the assay, these are like high content assay. You want to analyze thousands of samples in a go, hundreds of samples in a go, or something like that. And second thing is that you don't want to use a lot of material. These assays are done with 55 microliters of plasma. So think about like extracting, the patient samples are scarce. You don't get lot of material from the patient sample. So they want to actually use it as minimum as possible for this kind of assay, and then only this will work. So that is why they don't go to that path of extraction. It will work with extraction also. Okay, it's not that it won't work, but the idea is that you don't have to. If you do extraction, you have to spend extra money for extracting those things also, no?
But if you are comparing a cell, if you have a cell where you have the transcriptomic data and you want to compare the protein data as well for that cell, then wouldn't it be a good idea to have the protein from that cell?
Yeah, protein from that cell also. Like you take the cell, lyse it, and so lysate is that protein. You don't have to purify in that case. So you can work with lysate. So that is.
We are doing one project where we have transcriptomic data for some patients with SLE. So where we have done the transcriptomics of the PBMC, and then we thought that protein will do on the serum or what, but then it's not the same.
Yeah, so just lyse the PM.
That process we are doing.
Yeah, we can do that. We can do that. So these are now those interacting DNA and somamers, what they call as, they are actually now immobilized on a streptavidin beads. So once you do that, after that, what you do, I told you like there is a, if you remember this, there is a linker here. This linker, what you do, these are photocleavable linkers. So you shine UV light on that, and then it will cleave your complex that is actually bound on streptavidin beads away from streptavidin beads. So now everything is in solution. So you basically remove the beads and you take your solution out. and solution what you have is actually this complex, which is protein aptamer complex. Okay. Now you add A, and so now the other thing is, during this step what also you are doing, you are dissociating. When this cleavage happen, you are also dissociating any protein that non-specifically interacted, you know. So there are some low intensity, low affinity interactions are also there, which is non-specific. So that they will be actually during this cleavage, they will assure that those interactions lose the contact with each other. And so now the non-interacting proteins and your aptamers now separated, but those one which are specific will stay in the complex, and they will add something called as a polyanic competitor, which will go and bind to the free protein. So that means now these free proteins are not, they cannot come and bind back. So non-specific proteins which are in the solution, which are free of aptamers, when you add these polyanic competitors, they will come and make a complex, and they will stay away from any further interaction. And that means now you have a very unique complex which will with somamers. And
How these non-specific are released?
eh?
How these non-specific proteins are released?
So by when you are doing this, when you are actually really cleaving it out of your streptavidin beads through the linker dissociation, in that step the salt solutions are also changing a little bit. So then that will actually release any non-specific interaction, and non-specific protein will come into the solution, and their respective somamer also will come in solution. Then they will add a polyanic competitor that will go and bind with the free protein whatever is the solution. And then what you do, you actually go and add next biotin moiety into the complex, the aptamer protein complex, and you pull it down. After that you dissociate the protein and aptamer, which leaves a set of a DNA fragment. So basically now you have a set of DNA fragment, you make library and just sequence it. So those, and each of those fragments, short fragments of aptamers are corresponding to one individual protein, and you just do like RNA-seq kind of analysis where you say, okay, you count how many of those are coming from each of those molecules. So you are generating essentially same thing what Francis is teaching, that now this is not RNA, but what you are doing, this aptamers, you count how many, what is the count of those aptamers in your sample. And no, it is real quantification of protein. Yeah, relative in sense like it's all, they have always a standard curve, okay, and on that actually the quantification comes in. So in that way, relative means like you are actually benchmarking it against something, but that is not really they are doing. So this is how it is actually done. So you have somamers interact with your protein of interest, then you somehow think about all these things you are doing, and then finally you get that complex uniquely, and then you get the DNA out of that, and then make a library and sequence it, and then you count it, and then. So there is another technique called as O-link, which actually Dr. Amit was mentioning.
You want to examine multiple proteins?
Yeah, so SomaLogic can analyze up to 11,000 protein in a single lysate. So as 55 microliters of a lysate, you can analyze up to 11,000 protein. You can.
We need different type of aptamers.
So yeah, it comes as a mixture. So it will come like 11,000 aptamer mixture. Yeah, they have customs also, but yeah, but the highest size is 11,000. So it comes to the mixture. So you take the mixture, you add into your thing, and then everything happens. Yeah, 11,000 is the maximum. In case of this another company called as O-link, which also makes a proteogenomics kit, and this is a little more simpler than. And it's antibody based again. So any problem that you can think of antibody is there in this case, but the way how they get specificity. So what they do, instead of using one antibody, they use two antibodies. So and then each antibody will have a small DNA fragment attached to it, which is complementary to each other. Okay, if actually random antibodies comes and interact with it, they won't make the complementary pair. So if the right antibody comes and then recognizes two different domains of that same protein, then this will make a portion of that DNA will be double stranded, and they just extend that like primer extension you do in case of, and you extend that and make it whole completely double stranded molecule, and that double stranded molecule now you have this double stranded molecule, you just make a library and sequence it. So each of those again sequence can be traced back to the antibody, and then antibody can be traced back to the protein of your interest. So this is actually, this is how two different approaches, one is based on aptamers and one is based on antibody. Yeah.
Even in the Olink, they have basic version where they're using microarray.
No, Olink don't have any microarray based.
For the smaller numbers, they have a microarray, hybridization based.
So not microarray. So they have they are called as Q100 system, which is based on qPCR. So what they are real Taqman based qPCR probes. So once you release this DNA, now how you quantify it's up to you. Okay, so one of the way they do for the smaller panel for 48 panel, they are using something called as old fluidine super arrays. So those who are actually old enough, they know something called as fluidine super array. Super array means what happens that you have, it's a qPCR. So you have a 96 well this side of it, it's a glass plate, and 96 well this side and 96 well this side. And in this 96, this side of 96 well, you will add samples on all the 96 wells, and this side you will add primers with Taqman primers. So you can add all individual 96 primers. Then you spin your glass slide. What really happens that there is a center, there is a microfluidic chamber is there. So liquid will flow in all the combinations. So you will get combination of sample. So 96 by 96 combination you will get, and then so that means you are doing that many assays. And it will do the qPCR, and then you calculate the CT values and do the quantification, and so what Olink is using the Q100 system, they are using something like that. So it's a modification of their old fluidine system. But
So if these antibody which have DNA molecule labeled with them, so can these single-stranded DNA annihilate the complementary strand without the antibody binding with the antigen?
no, because of actually so many molecules are there that it's hard for them to, because so DNA affinity is it's based on affinity. Okay, so DNA affinity in a liquid system is actually lesser than antibody protein affinity. So any reaction kinetics is actually driven by that affinity of a reaction. Like so whichever is having a higher affinity, they will actually reaction will tend to actually go to that direction. Okay, so it's not that free DNA cannot actually do a reverse complementary. It's just because the favoring reaction here is antibody protein reaction, and so that is why most of the molecules will be
Still there may be some free antibodies available which are
less. It's very less. Actually, it's so crowded environment that actually it's very less. That is, and then the salt optimization and all will give you that balance, and that is the innovation here actually.
They call it
Proximity ligation, yeah, that is what actually it is called as.
maybe complementary bases are less.
No, it will not, very few. That is the innovation there. Okay, that is how the whole reaction is actually optimized in a way that the just antibody based pairing should not happen, and that is done through controlling the salt concentration in the solution. Okay, so we'll see a little bit more time anyway. We are, if we could finish it then. Okay. Anyway, so one of the major application, and why all of us are here, because we are interested to use NGS for the clinical application. So there are various type of variants you have which actually can be identified through NGS based approach. Like single morph SNPs are there, insertion deletions, repeat expansions, frame shift mutation, defective splicing, or even the premature codon insertion in the genome, okay, in the genes. So what is actually the purpose of DNA or precision medicine, DNA sequencing and precision medicine? It can be for discovery, it can be for diagnosis, it can be for classification of disease stage, it can be for prognosis, it can be for therapeutic choice. So it depends upon what is your application. You can actually tailor your NGS based analysis for, but the problem here is that sequencing a genome is pretty simple, and and as I mentioned you, like we are sequencing every day and 30x genome, almost 200 250 genomes every 48 hours from our facility. So sequencing is not a problem. It's simple, and now it's not expensive too. So that means simple, inexpensive technique. But finding a genetic variant is not. That is where the whole problem is. So it's basically you have like millions of single nucleotide polymorphism in each of the individuals, and some of the effects that each of these variants shows are more cumulative in nature. It's not like, you know, it's a and the term that we actually like to use is called as penetrance. So we don't know what is the penetrance of a variant, and then if unless it is 100% penetrance, then, and most of the variants they are not have that kind of a penetrance, and only variants which have those kind of penetrance are the one which are on the enzymes. So if you have a SNP which is within the enzyme, and it's within the enzyme active pockets, like best example is sickle cell anemia, where you have that one SNP is actually valent to isoleucine SNP is in that hemoglobin binding pocket, and that means every single individual with that one SNP will actually give rise to sickle cell. So if you have a condition like that, which is mostly in case of biochemical enzymes that are active in the system, but apart from that, if any other transcription factor or any other kind of component of various component cells, you will never get that kind of the penetrance in the population. And once you don't have that kind of a penetrance, then it's get harder and harder to classify their importance in the. Okay, and essentially you, Dr. Amit yesterday went through this, but then the idea here is that you can classify variants based on what kind of evidence is present in this, and then based on that you can use them for various purposes. Like you can use variants of strong clinical significance like tier 1 variants, and they can be, and for that those variants are used for therapeutic, prognostic as well as diagnostic purpose. And similarly, actually you can also use tier 2 variant, which is for these purposes. Again, they need also, you know, various level of evidences. But one of the most challenging one for this is basically for somatic variants, but then tier 3 variants, they are the most interesting ones, but that is when we don't have any handle to deal with, and they are variants of unknown clinical significance. And most of your patient samples, you will see a lot of them having a very strong association with the phenotype that you are seeing, but you have no idea why you are seeing them, or you need an extra layer of experimental evidence to prove that this. Like one of the examples that I can actually think of, that we had a patient sample where we got a SNP in NeuroD, and this boy, it's a 14-year-old boy, and he has seizures. So, but then there is no way to prove that, or there was no literature even available that particular gene, NeuroD gene, any mutation in NeuroD gene has any seizure phenotype. So now the question is, but that is the only association that we can see. So now what we need to do. So what the group, our pediatrics group did, they deleted that they created the same deletion in zebrafish, and they found that zebrafish show the same seizure phenotype. So, but then think about creating such a line of zebrafish and then testing these phenotypes will take months. And when you are doing this for gene after gene after gene, or SNP after SNP after SNP, it's actually gets more challenging. So though this is the most interesting group of variants that you will ever see, but they have there. And then your analysis will show you large number of variants which are likely benign in nature, and we don't know why some of those variants are, and then some of them shows very strong association also with them. Okay, so while we can use some of the SNPs for classification, but one of the strongest area where we can use some of this NGS based method is for pharmacogenomics. So old way of doing medicine in India, and especially in Arabic countries also, they have used and they have treated each human being as an individual human being, and they classify human, and in traditional Ayurvedic medicines, they are classifying human being as Kapha, Pitta and Vayu in their nature. But actually I think that is, maybe a Kerala term for that. So what is really happening? You are not considering all human being as uniform in nature, and then treatment, you are treating not the, you are not treating disease, you are treating that person, and each person need a tailored approach to treat. But when modern medicine came, and especially evolution of modern medicine was mainly for pathogenic diseases, especially like antibiotic was the first major thing happened, and it was highly successful, because at that point you are not treating, and you don't have to treat a person, rather you have to treat a germ, like you have to kill some germ, virus or something like that. So it's highly effective in that case. But when it comes to lifestyle diseases like diabetes and hypertension, we have to go back to that old idea of treating a person. And why we need to do that? Because of this, like each human being has a different based on the polymorphism they have in various genes, they will show a different pharmacokinetics and pharmacodynamic properties against a drug. Okay, and so basically, what is pharmacogenomics is a study of inherited differences in inter-individual, like between individuals, about drug disposition and its effect. So same drug given to two different people will be reacting two different ways, and we are actually studying how this is happening. Right? So pharmacogenetics is basically looking at the genetic variation and how that is interacting with drug, and pharmacogenomics is basically looking at those in a population, and giving you a idea that how the population in general is behaving, and what are the multigenic determinants for the drug response. Okay. So one of the things that we know all the time, that one doctor prescribes a drug, and then it's actually effective for some people don't respond at all, and some people actually has serious side effect. The same medicine actually given to the 3 different people, and the question is like, why that is so? So this difference comes from 2 different aspects. One is called as drug uptake, and second is from the drug metabolism. So how the drug is taken by each individual and how that metabolized. So if your cell, the cell that actually taking the drug has many receptors, that means what happens? You have the drug, and all the drug will go and bind to that cell, and it will have a very strong reaction. Okay. If you have fewer receptor for the drug, you will have weaker reaction. If you have no receptor, the drug will not work. It will be just you are just swallowing the pill. And also, if you have a different receptor than the receptor that you need to have, in that case also your drug will not work. Okay. The same thing, the drug uptake also, if actually drug is normal uptake, then drug will work as it is actually intended to work. But if there is a decreased uptake of this for some reason, like you have some channel issue in your cell, then it may build up in your extracellular space, and that will create toxicity. The other aspect is like how drug is metabolized. If drug is actually metabolized at a normal rate, then the normal drug will respond to the normal response. But if it actually it breaks very fast, then you need more drug. Like, so you need a higher dosage of that same drug to that person. And the third thing, if the slowdown, it's a slowdown, there's a slow breakdown, then you need to reduce the dose. So we need to know all these things. And I will show you some few examples where, and all of this aspect what I have just showed you, it's controlled through the genetics. And that means the SNPs in some of the genes that will define the response of those drugs either at the metabolic level or at the level of intake. So this is about a medicine, abacavir, which is actually a reverse transcriptase inhibitor used for the AIDS patient. Okay. So the patient, whoever carries this allele, HLA-B5701 allele, they are at very high risk of hypersensitivity. So now what they are doing, they put this in the medicine label, that if you are prescribing this drug to a patient, first test for this allele and make sure like you don't have this, and then if the patient is positive for this allele, then go for an alternate medicine. So this is one such example where you are really looking at a SNP and making sure like if that SNP is not there, then only you prescribe that medicine. You cannot otherwise prescribe this. The other example is actually warfarin. Okay. So I can show you many such examples, but this is another example where in case of warfarin, if the variation is there in two different genes like CYP2, C9 genes, or VKORC1 gene, either of these genes have a SNP, that means you should reduce the dosage level. So first example we have seen that you cannot use the drug for that patient at all with the presence of that SNP. But in this case, you are actually changing the. So both examples we have seen. In one example, we are completely excluding that patient from taking that medicine, while in this case, you are actually need to lower the dose. So based on your pharmacogenomics output, you will be able to tailor your prescription. You can tailor your dosage, and then you can come to. So any of these things now actually, I will go to CPEX little bit later, a couple of slides from here. There are recommendations for various genes and what is that gene SNP relation with the medicine and what you need to do with that, and we'll look into that later. So what are actually the challenges for implementing pharmacogenomics? So one of the biggest challenges that translating this genetic information to clinical action. So now we'll see like there are fewer genes only those who are reported with such things. It's not that that interaction is only for those genes. It's just because it's not studied well. Then again you are adding an extra cost, because if you need to do this testing, that means you also somebody has to pay for that. And then we need to have some kind of a genotypic testing, high quality genotypic testing, and some of these things are actually more related with US, but again, the practical challenge is to translate this information, genotypic information into a pharmacogenomics information. Okay. There is a consortium called as Clinical Pharmacogenomics Implementation Consortium, which is called as CIPIC. Okay. So the whole idea here is they want to actually standardize this format, report guidelines format, and then they want to actually provide a freely available peer-reviewed data for pharmacogenomics. And this consortium is an international effort. And what they do, they actually publishes the drug and which SNPs or the gene that you need to test. So up to 2021 data is available in their website. I haven't seen after that. So each of them, so you can see like each year they have new drugs, like in this case CYP2 D6 for tamoxifen or something like that. So they will actually tell you that even if any of this drug you are prescribing, you should actually test for that gene before you prescribing and look for the SNPs that may be determinant for or to decide the dosage. So based on that, and they have a detailed guidelines for each of this medicine, and now it is actually for other. This is just a list, and you can go to that website, and then you can read more about this. Okay. So what is the benefit of one of the major benefit is like you can prescribe drugs more effectively and avoid prescribing over prescribing a drug. Okay. And then also, you can also make patient adhere to the practices that you want to do and the right drug. It will improve the life quality, reduce the cost, because you gave a drug which is not working in that patient means like the patient will be having that drug for a couple of months, so he already wasted a lot of money on that and time, valuable time that is lost. And you can also revive some of the drugs that failed the drug trial, because it might happen that they need to test that population for those SNP and the drug failed just because those things were not considered. So you can actually revive some of them, and of course you can control the cost. Yeah. So what are the potential legal and ethical issues? One of the major ethical issue is that who should have access to the person's genetic profile, because you are doing a genetic testing, so who will control that data? How will the protected genetic privacy and prevent genetic discrimination in the workspace and in our healthcare system? So I usually try to joke when I teach in US that you know, in India, we usually have arranged marriages, which is not much known to. And if somebody has access to my genetic data and say like I will have a heart attack at the age of 40, most of the people will not give me a girl for me. So, so that is so the whole idea is that how you can maintain the privacy of the data and then who will have access to it, okay. And other thing is like your employer. Again, the private companies and all, when you're going and they know that, okay, you may have a tendency for depression or something like that, will they take into account when they are giving you a job? So all these kind of issues like privacy related issues and who will have access to that data, and even insurance. Now insurance is not that popular yet in India, but then it's getting popular. So insurance will classify this as a pre-existing condition. So will they pay you for some of the disease? So all these things actually is important that we have to think about when we are, again, this is more to do with US, but then one of the areas that which we have a real problem is this integration of pharmacogenomics with the personalized medicine, and marry both of these things and make clinician aware of use of it, okay. So now we are actually coming to the last part of my talk, which is we are moving to the 4th generation sequencing, and Dr. Amit actually already gave you a primer for this, that single cell analysis. The 4th generation of sequencing has, it's not a new sequencer, rather than it's more about a new way of doing things, okay. So what are you doing here? You have a tissue. In bulk analysis, you just make a smoothie out of it, and then you analyze it, and then single cell analysis, you will analyze each cell separately, and you get the data out of it. So why it is important? Because you know, if in India you go to a smoothie shop and you have a smoothie or mixed fruit juice, a lot of places have very popular. So the taste of the mixed fruit, all mixed fruit juice is like, I'm from Hyderabad. Those people who are from Hyderabad, they will know. This is a very big delicacy in Hyderabad, all mixed fruit juice. So the taste of all mixed fruit juice will be according to the what is the cheapest fruit in that part of the time, because and most of the time it will be banana. So you won't get even strawberry taste or anything. So essentially what I'm trying to say here is that in bulk RNA-seq, the flavor that you get out of your data more or less coming from the dominant cell type or dominant the phenotype that you are. But the whole idea is that if that is what we are looking for, it's fine. At the end of the day, we just want everything else a little bit, but then something dominant, we are fine with that, then it's okay. So that one limitation is something that we can actually overcome using single cell RNA. But the why we actually need this? So one of the best example is cancer. So we always think when we think about cancer, every type of cancer we think as a homogeneous mass, like something proliferating, and then everything is fine. But that is not the case. And the main thing what we learn over a period of time is that it's more heterogeneous. The cells, they are in the core, the center of the mass, cancer versus something that is actually exposed to the periphery. And those who are nearby the vascular system, like where the angiogenesis is happening, they all are different. So which means that there is an inherent heterogeneity, and that is why a single drug cannot target all the cell in a even the chemotherapy and all, and you still left with some cells in the system. Okay, so if you want to design a drug or a treatment strategy, you need to understand the heterogeneity of your tumor, and you need to actually address the heterogeneity part of that, so that all the cells will be equally targeted, or at least you have a combination of approach that can actually target everything. Okay. So knowing heterogeneity is important, and same thing actually goes to various other areas where when you are looking at tissue homeostasis, or you are looking at what are the different cell types in the tissue and how they are actually interacting at any given point of time. So heterogeneity is the rule in biology, and without taking that into account, we cannot actually make sense out of the system. So to understand heterogeneity, we need to do single cell rather than going for the bulk RNA. So this is how actually it is. You take something like a PMBSE or anything you are taking, you can basically generate the catalog of all different cell types, and also you can actually get some sense of the population dynamics of those cells, how they are changing their nature. So, single cell RNA-seq can be done with 3 different strategies. Okay. So, one is called as single barcoding, second is called as split pool barcoding, and third is through microfluidic space system. Okay. And I know that you all are familiar with all 3 when I actually show you what it is. In single barcoding, what you do, you sort your cell into one cell into each well and add one barcode, do an lyse the cell there, do the RT-PCR. In that RT-PCR step, you are adding one index into that. So, that will give you unique identity to each cell. And then you just after that doing that RT, you combine everything and then do a library prep and sequencing. So, then what is happening, each cell has a barcode. So, the RNA that is coming from each of those cells will have that barcode. So, you use that barcode and then just deconvolute later on. But the problem with this approach is this is very low throughput, because you cannot analyze more than 90 or 100 cells or 200 cells through this method, and it is expensive also, because you are doing one well at a time, which makes it very expensive. Okay. But there are systems like iCell8 and those kind of systems are there, which are essentially doing symptoms similar to this. Okay. But this is again, this is not the way single cell technology is going. We are not actually not much into this kind of approach, because the cost becomes cost prohibitive. Yeah, it started this way, but now it's not that popular. For the historical context, I thought it will be it's a good idea to start this way. So, this other thing what we do is called as split pool barcoding strategy. Okay. So, split pool barcoding, what we are doing, if suppose you have you want to analyze 10,000 cells. Okay. Now, you pool 100 cells and split them into 100 wells. So, you have 100 cells, you split them into 100 wells. Do first RT. So, each well has a barcode. So, that means all the 100 cells will get one same barcode. Now, once you have barcoded, you have did your first RT, you pool every cell back, all the 10,000. So, now 100 cells in 100 wells, you pull it back, you have 10,000 cells back into one pool. You split it again into 100 cells. So, now and then put it into 100 wells. So, now each well will have a different combination of cells. You add your second barcode. So, that means each of those 100 cells you got a different combination of barcode now. Then you pull again and then you split again. And if you do this split pool barcoding 3 times with all the available barcoding, you will generate approximately 100,000 combinations. And that 100,000 combination you will actually and then pull everything and then make a sequence. So, basically what you are doing, you will each of this cell will have this, like you have this is of like 4 set of barcode, and this also have 4, but you see the order of each of these barcodes are different. So, it is the combination of barcodes, also the order of those barcodes are also different. Yeah. So, this way you can create any number of combination through just by splitting and pulling. So, what is the advantage of this system is you do not need any specialized instrument, because it is just pipetting based. You have, say you have 100,000 10,000 cells in 1 ml of media, 1 ml of media, you actually split them into 100 microliters into each, that 10 microliters into each, that means each of them will have 100 cells.
How do we extrapolate which cell is this? Suppose this is monocyte or this is...
That is based on your transcriptome. So, you will be, so it is only the barcoding part I am talking about.
What is this barcoding actually?
Barcoding is actually, so you have a RNA, okay, you are putting a tag in front of that RNA to identify that. It's a string of nucleotide only, but it is actually, so you are putting that in front of an RNA, and if you have say 10 different genes coming from the same cell, all of them will have same string of DNA. So, then you will, so the first thing is like based on that you will combine all your RNA data into and say like this is coming from cell number 1, this is coming from cell number 2, this is coming from. Yeah, they already have that. So it is called as.
How does it compare with?
It is good. We actually started offering that from our service. Called as. So we. Okay, I will come to that. Just let me finish this and then come. So this is actually called as. So this is called a split pool barcoding strategy. The whole single cell analysis is about barcoding. How you barcode a cell transcript. Okay. So first way of doing is like you pipette cells. Second way is like this split pool strategy, and you create a combination of barcodes inside each cell. The third one is through microfluidics, which I will be saying. Now coming back to the PIP-seq. So we were the lab which was providing this for them. So they were providing initially services. A company called as Fluent who generated this system called as PIP-seq, and it's again based on split pool barcoding. But now Illumina purchased that platform. So we were the one who was sequencing from last 2-3 years, and we are offering that as a service from YCGA now. So it's simple. You actually take cells, put into a mixture of things, vortex it. They have a vortexer, and then you spin it down, and then they have some pipetting things. That's it. So it's very straightforward and it works well. I will actually send you a paper. There is a benchmarking paper is there. Our own benchmarking also shows that it's comparable with the 10x. So it's comparable to 10x data. Very cheap actually. It's almost 4 times cheaper than. What specificity?
She's asking me if you are putting 3 or 4 tags in front of one piece of RNA.
No, so you are putting them one at a time. So that means that combinatorial. And each of them, yeah, will be having unique combination. So that is how you get that specificity. No, it has nothing to do. It's way before monocyte or anything. Forget about that. It's just cells are cells. Okay. What we are, whether they are monocytes or not, that information will come from the gene expression data, not from the barcode data. Okay. We are just saying cell number 1, cell number 2. So you need to identify that if you are sequencing a piece of RNA, whether it is coming from cell number 1 or cell number 2 or cell number 3. So to classify that, you are doing this barcoding. Okay. And once you get that, then you have the RNA. So you will get all the RNA coming from cell number 1. Now you go and classify that cell based on the RNA content, that whether this is a monocyte, this is a T cell, native T cell, CD44, all these things.
But within a seemingly homogeneous population, even like a monocyte, there will be different types.
Yeah, there will be different types are there. All those, so you can do all those things. So essentially, the barcoding step is to barcode a cell.
So
And
barcode is cell. How can these specific barcode be added inside a cell?
when you do RT-PCR, that is what you're doing.
So the cell should be live.
So in the case of
Yeah. So at some stage they are actually doing the lysis. Okay, so in split pool strategy they usually what they are doing, they are actually lyse at the last, but then the reagents can actually go inside the cell. They are made permeabilized and then they go inside the cell, and you can do in situ RT-PCRs. That is what actually essentially they are doing in this case. Then you don't have to lysis happens actually. Lysis happens after all the barcoding steps are done.
So cells are initially permeabilized and then they go inside and
Yeah. Yeah, because you need to maintain the integrity of the cells at that point.
So we should know the cell number. Is it specific to the cell number, the barcode?
No, so parse, what Mr. was talking about. So parse is actually you can do, the current kit you can do up to 100,000 cell barcoding. They have a kit for that. So yes, all these barcoding kits actually they comes with a for the specific number, like they have 10,000, 20,000, 100,000. So you have to, based on what number you are targeting, you need to actually start with that kit, and there is always a recommendation for how many cells. So if you want to capture 10,000 cells, you cannot start with 10,000 cells. You need more than that. So like PIP-seq, you need at least 2 times more than that. If you want to capture 10,000 cells, your input cell number will be 20,000 cells, and then only you will. So capture efficiency, what the term that used here is called as capture efficiency, and for 10x it is around 65%. But for other like parse and fluent, it is actually around 50%. So based on capture efficiency, you need to extrapolate. So if it is something having a 65% capture efficiency, that means if you want to capture 10,000 cells, then your input will be 17,000 cells. If something has a 50,000 capture efficiency, then you will have to have a 20,000 cells. So like that. So and then again there is another thing actually. I just okay. Let me finish it, and then I will come back to another round of discussion on something else. So in case of microfluidics, again the idea here we are still barcoding cells, okay, and then we are doing rest of it. So in this case, what we are doing, there is a bead, okay. This bead consists of a adapter, and each bead will have a unique barcode. Each bead will have a unique barcode, and then some other portions will be there. Then what you do, you send it through a microfluidic chamber, and it has 3 inlets. From one side your bead will be coming, from other sides the reagents and beads will come, other side cell will come, and from the third side the pressure inlet and oil is actually going, and a pressure is exerted. In this process what happens, in this junction where these inlets are coming, they make a oil droplet, and this pressure is controlled in such a way that each oil droplet will have one bead, one cell, and all the reagents required for RT-PCR. And then this, what they do inside that oil droplet, it's like an
Not be the case actually. I mean, like a lot of cells, a lot of droplets will be there where there might not be cells, where there might be 2 cells.
yeah. No. That's where I will come to that, like how that is done. So basically, as a workflow, what happens that you have. So idea is that you get a oil droplet, and each oil droplet will have one cell, and each droplet should have some reagent and one bead. Okay, and now the reagents are there in this. They will lyse cell inside that oil droplet and do an RT-PCR, which will be so in that RT-PCR step, the primers are provided by that bead. So that means it will insert a barcode into your cDNA. Now after that, what you do, you break open the oil droplet. Okay, you add a detergent that will break open and then pull your whole RNA now together and just sequence it. Now what Amit sir was talking about, the innovation of a technology is there are 2 issues in this case. One is called as doublet rate, where you are actually having 2 or 3 cells going into single oil droplet, or it never happens. Actually, mostly it's 2, 3 rarely happens. It's actually, if you are that bad in you're experimenting, then problem. So and dropout rates. So there will be some oil droplet where there is no cell. Okay. So either of these conditions. So one of the way it is done is through controlling the cell number that you are, and if you have a good suspension, you have cells are single cell suspension, and they are not sticky or anything like that. That is how it is done. The second thing, the whole innovation is in the pressure, the control of the pressure by the instrument, the chromium system or anything. That is one of the other factors here. But the third thing, which is basically relevant for doublet rate, is that more cells you analyze. Like say if you are analyzing 5,000 cells, you will get fewer doublets. But doublet rate increases exponentially as you analyze more number of cells per channel. So suppose so the doublet rate increases to almost 7% when you are going from 5,000 to 10,000 cells per channel. So, but then there are bioinformatics way. Hopefully, Francis will be showing you that how to remove the doublets based on. No, you are not showing that filtering. Okay, so you can actually bioinformatically you can filter the doublets from your data, and then you can clean up it for that. So that is how the doublets are taken care of. Okay. Yeah. We are just introducing them to this. So again, why single cell? Basically, we are cataloging different cell types. We want to identify new cell types. We want to identify differentiation trajectory or dynamics of any developmental process. Yes. Okay. But what are the biggest challenge for single cell analysis? One of the biggest challenges, it's big data set. Okay, it's so big. The other challenge is shallow sequencing. So most of the samples are sequenced at, say, every cell is sequenced for 20,000 reads. So that is the minimum sequencing. Some people go all the way up to 50,000. But think about this, on an average, 1,000 genes are expressed per cell. That means you only have 2 or 3 reads per cell, per gene in your data, and how reliable that shallow sequencing is. So this is something that you always think about when you make quantitative inferences from single cell data. I always try to argue with people that single cell data is non-quantitative. It is more like a qualitative data. So please treat that as a qualitative data. Don't use that for gene expression. And then because of that, like you are shallow sequencing, and each cell has its own variability, the technical variability as well as biological variability of data is very high. Okay. So one of the best practices, because of these issues, because of high technical variability as well as the biological variability, do not confound your experiment by batches. If you have one set of experiment you are doing, don't do them in batches rather than do everything together, because that will make it easier for you to. And if you are actually doing in batches, then split your replicates in batches. So don't do like for one set of sample you do all replicates today, and then other set of batch you do other replicates. Rather than that, split across the sample replicates in different batches. That will also help you to, and add as many replicates as possible. And this is easier than said. The biggest problem is the cost. So even like RNA-seq, people are reluctant to add more replicates. Here actually you are talking about 10 times more cost than the bulk RNA-seq, and so it's a harder thing to do. And also whatever actually Francis told you about the RNA-seq, it's also relevant that always add metadata or collect all the metadata about your experiment, and that will help us to understand the batch effects. So technical variability of most of the RNA-seq experiments comes from cell-specific capture efficiency, library quality, amplification bias, and the batch effects. So one point I want to actually emphasize here about amplification bias. Why amplification bias happens? Specifically, it was mentioned in not the bulk RNA-seq, but it is being mentioned in case of single cell RNA-seq. The reason why this happens, because you are working with a small input material. Any small input library prep, DNA, RNA, all of them will show some level of amplification bias. Okay, and we see a dominant effect of amplification bias in single cell libraries because of that. And the source of biological variability comes from transcriptional bursting. Anybody know what is this transcriptional bursting means? I mean, none of you are molecular biologists, okay? So I'm happy that I can even say something wrong. Okay, so what do you mean by transcriptional bursting? So tell me, when you think about transcription, what do you think? How it happens?
As long as the replication is going, the transcription is getting size.
So that is what I also thought. Okay, that your RNA polymerase comes and transcribes, and then another polymerase will come and transcribe, and another polymerase will come and transcribe. But in reality, that is not how it happens. It happens in bursts. So polymerase will come and engage for 30 seconds or 1 minute, and it will transcribe everything, and then everything is silent. Okay, then like after 5 minutes, again it will happen, and it will happen again after 5 minutes or 3 minutes or 2 minutes. So it happens in burst, and this is called as transcriptional burst. If you actually ever quantify a nascent RNA on a microscope based method or imaging based method, you will be able to see that transcriptional burst happening. So the idea that transcription happens as a continuous process is not true. Transcription happens in burst, and then burst that is precisely controlled process. So
Single cell will not be able to see that burst.
no, that is the problem. The single cell, it actually taking that snapshot more effectively. In the bulk RNA-seq, you are actually normalizing that because you have millions of cells, and now say if you have a million cell in your soup, 10,000 cells will be bursting, 20,000 cells are not bursting. So you will not be able to see the effect of burst, but in single cell, you are picking that one cell, and then you are actually putting it through your active. So that is where the cell is same. These two cells are same, but one is not bursting, one is bursting. Then it will be seen as two. So that is the source of biological and you are. So every cell, every gene do that. That is how the transcription happens for every single gene. That is a mechanism. So single cell is actually able to capture that. That's why you see a lot of biological variability in single cell data compared to bulk. Then again, rate of RNA processing. So each gene actually processed at a different rate. So the one which is processed slightly earlier versus late. All this dynamics of transcription and processing is captured through the single cell.
Even for genes where there is only slight difference where they can pick on bulk, the difference would it be exaggerated on a single cell?
Yeah, and it has to be controlled. So the whole this biological variability thing coming from singles. Sometimes you see clusters, cells getting clustered into two different clusters, and you see more or less their general expression profiles are same. That is coming from this kind of changes. It's not a real difference. Rather than it's more like coming from all this kind of thing, which is actually I believe giving rise to the biological variability. Okay. So then the single cell is good, but the biggest challenge, like we started with heterogeneity. Okay, the whole idea of heterogeneity, just knowing that you have a cell doesn't matter. You need to know where is that cell, what is the position of that cell. Okay, and think about a neighbor. You have a neighbor who are like very rowdy kind of a person. Like or the morning, everyday morning. Or you have a house next to your house, and husband and wife is fighting every day morning, and that is what you are hearing every day when you wake up. So your days are spoiled because of your neighbor. It's not because of you, but because of your neighbor.
I actually don't live close to
and same thing, but think about a very nice person. Morning you actually come out to pick your newspaper, and this guy say, "Hey, hi, how are you doing?" Well, that makes your day. Huh? So neighborhood is actually one of the key determinant for disease development, everything. Cells, those are close to vasculature, they behave differently than those which are far from the like blood vessels. Or cells, those are in anaerobic environment, not anaerobic actually, but oxygen related term, I forgot that. What is that? Yeah, whatever. Yes. So basically, the area where you have less oxygen tension, it's not hypoxic. Actually, they have anoxic or something. It's anaeroxic or something. That is the word. Basic idea is that wherever you have less oxygen tension, cells behave differently. And you are further from the vasculature or the vein or the artery, the tissue will also behave slightly differently. So you need to know your neighbor. And knowing the neighbor is important. And that is when you actually do spatially resolved single cell microgenomics. This is a word that it's a right word. When we say spatial technology, rather we like to say spatially resolved single cell sequence. So you want to see single cell data, but you also need to have a spatial information along with the data. Okay, and you can do that two different ways. Something like called as sequencing based, and other is actually imaging based technology. Oh, I know why my slides are behaving this way. I have to change my resolution of my laptop. I don't know. That's okay. Anyway, so sequencing based method, what you are doing, you have somehow you are barcoding you are including two barcodes. One barcode for the cell, because you need to identify each of the cells separately, and then you also need another barcode to identify the relative location of that cell to compare to each other. So you need to introduce those two barcodes, and then you do a sequencing. But then also you can do that based on imaging based approaches. So example of imaging based technologies like Visium, Cosmex, Xenium, like that. And if you are thinking about sequencing based, you have again Visium or Genomics or Curio. So some of these platforms give you, and we'll see some of this quickly what it does. So the biggest challenge in the spatial technology field is the trade-off between resolution and number of targets, because these are expensive, and if you add more number of targets in your analysis, it will cost you more. Okay. The workflows for most of the spatial technology is hard, it's complex. And you need to optimize each tissue, especially spatial imaging based spatial platform. You need to do clearing, because there will be a lot of autofluorescence coming from the tissue, and you need to have a means to clear them. or the permeabilization protocol. So, each of this tissue for barcoding, effective barcoding, you need to permeabilize tissue properly, and each tissues with based on their fiber content as well as like how much cartilage or bony contents are there, based on that you need to actually change your clearing as well as permeabilization protocol. Then high assay to assay variability. So, this is the field newer field, and we do not know what is the real source of this. It may be biological in nature because you have no means to try same tissue section 2 times. So, we do not know that is a technical variation or is it coming from because if you are taking 2 sections, they are not same, they are actually different because they are 2 different layers. So, that means we do not know where this is coming off. Again, the assay cost is a big thing. The other issue is, are you actually interested to do discovery? Discovery means like you need to test as many as genes, or you want to have a focused question like neuro-oncology panel for cancer or something like. So, the whole idea is a discovery versus focus question. Then it actually is expensive. These instruments are expensive, so you need to spend additional money for capital equipment, and definitely there is a big bottleneck in terms of visualization, data analysis and those kind of things, because there are not many people who are expert of doing. Okay. So the first very popular what is happening? It copied multiple times. Francis, what is happening? This is your laptop, it's not behaving, it's like you, not behaving properly.
No, it's the other people scrolls in a different way than everybody's wrong except me, that's what I'm thinking.
My boss actually used to say that.
Why? You don't know how to scroll in the other way.
I'm not, oh. No, it's not going anyway. See, it's coming back. What to do? Something is.
Something going on. What did you do to my computer? Are you pressing it?
They could finish it? Done?
Some people it works, for some people it is working.
It still is not running.
For some people it has, but not for everyone.
Not for everyone. I sent an email to AWS. Yeah, allow me to check.
So, okay. So, the first platform which is it's called Curio, and I want to, this is a sequencing based platform. So, what are you doing here? This is a small slide, okay, and on the small slide there is a lawn of oligos which are barcodes, so spatial barcodes, okay, and then what you are doing, you lay your section. It's actually 5 to 10 micron sections. You lay your sections, and mostly we just got the FFP protocol, but it was earlier just the fresh frozen sections, OCT embedded sections. You put the sections over that area here. So this is a lawn of oligos, and this consists of this set of barcodes there and a bead. Okay. Now you put your sections over that and put your finger at the bottom of the slide. The idea is that we have a body temperature of about 37 degrees centigrade. So we just allow that section to melt over that region. So just do that for like a couple of seconds and then melt it, and then you do a RT-PCR on, and there's a formalization step, and do RT-PCR, collect your RNA and just sequence it. It's a very simple, straightforward technique, but so this is actually called as curio. Okay.
But how many genes can it use?
It's the whole transcriptome. So it comes in two different variation. You have either you can analyze 3 by 3 sections, 3 millimeter by 3 millimeter section, or you can analyze 10 millimeter by 10 millimeter section. And the advantage, because this is sequencing based, so you are sequencing whole transcriptome, you are analyzing all the and the beauty of this method, which you will understand why I'm saying this, because you can, it's a species agnostic. Means you can analyze any species using this, because it's not looking at it's like a sequencing method where after sequencing you align it to the reference genome and then you figure out that what it is. Okay, so this is curio, and here is an example of data. This is a retinal sample from mouse retina, and you can see expression of two genes. One is this GFAP and Rho. So Rho is basically present in photoreceptor 1. Yeah. So that is actually present in photoreceptor 1 layer, photoreceptor layer, and you can see the expression of that in your retina, and also you can see GFAP expression in optic nerve head. So you can really see a granular data, but the resolution for this method is 10 micron. So that means if a cell is less than 10 micron, and then it will not be actually able to resolve those cells. But then this kind of analysis is very useful for neighborhood characterization and general understanding of tissue. They came with a new technique called as tracker, the curio tracker, and which gives you a data at single cell resolution. And at this point, in all sequencing method, curio tracker is the only platform which can give you single cell resolution spatial data. Okay. I don't know what happened to my slides. Actually, everything got messed up. Okay. So next one is an imaging based technology called as Cosmex. So this is developed by a company called as NanoString, and the way this assay works, you create a custom set of probes for your whatever number of genes you want to analyze. Say you want to analyze 50 genes. So you decide which 50 genes you want, and you generate the probes against those 50 genes. So, okay. And the probes are usually 20 base pair probes, and this probe will go and bind again, hybridize against. So you have a slide, and then you go and hybridize that probe, and then what happens that you do something called as amplify the signature using branched PCR reaction. So it's actually every PCR cycle it will add one extra floor. That is what actually happening, and so that is how you increase the concentration of floor, and you do this in a combinatorial manner. So you have multiple probes for each gene. So there are 5 probes for each gene, but all 5 probes you will not add in each cycle. So each cycle you will have a set of probes for set of genes, and then you are actually hybridize it, do the amplification, take a picture, wash it off or quench it, and then you actually come back and you add next round of it. So that way what happen, you will, so why you are doing this? Any idea why this way? You can actually just add all the probes together. Why you are doing that? Any idea? Because these are 20 nucleotide probe. The chance is that they may be binding to a non-specific target. So how you will get those specificity? If you have 5 oligos and each of them added a different cycle, and at the same spot they illuminate, that means you know that that is a specific one. But in random one, sometime you get a signal, you omit them from your analysis. So that is how you generate the specificity here. So this is called as Cosmex. Cosmex is the only imaging platform at this point which can give you whole transcriptome data. So, this is how this works. Now, again they have a slide, and this area you can put as many samples as you want, and then you can see localization of genes at subcellular levels also. You can actually see that because they are using DAPI, and there is another idea in spatial technology called as segmentation. So, cell segmentation is a very challenging problem in spatial technologies. Cosmex, they uses a mixture of 5 cell surface markers, and based on their presence they actually mark the boundary of cells, and then they use but they also use a computational approach to mark those boundaries, make sure that all the cells. So, some cells and then they use DAPI. So, one of the biggest challenge, like think about cell like muscle have multiple nucleus. So, that means if your DAPI stain is coming, that will show you a string of, but none of those DAPI stains are actually able to show the boundary of cell, because the cell boundary may be far off. Same way like neurons have a soma, and it has like all the body going all the way to like kilometers. So those kind of cells, it's sometimes hard when you act, some of the computational programs are hard to work on, but the things are improving, and so segmentation is one of the major challenges for many of these technologies. Okay. Other technology which is from 10x called as Xenium. Here they use padlock probes. What is a padlock probes? So, padlock probes, it's same thing what we are talking about in case of Olink we said. So, you have instead of antibody you think about two oligos, 2 primers. They are at proximity to each other. So, they will go and hybridize, and they will bind next to each other, and there is a DNA molecule having complementarity to each other. So, that means you will actually amplify that, and each amplification step you add a fluorophore. So, when that amplification happens, that signal will be coming out, and that signal you will capture through imaging platform. So, the specificity comes from 2 probes each gene they are using, because then what is happening, binding 2 non-specific fragment with the same complementary DNA is actually not possible. So, that way you will get a very high and here actually before doing the amplification, they padlock probes, they ligate to each other and make that circle structure, and they do a rolling circle amplification. That is how they achieve this. And here again this is an example how mouse brain, this is our own data, where you can see the position of various cell type in a tissue, as well as you can see their single cell distribution. This is a UMAP, a TSNA plot, and through this you can actually see various cells happening. And this is one example of how you do a computation based segmentation. So, here they are not using cell surface marker, rather than what they are doing, they are looking at the DAPI signal, and then they are expanding the DAPI signal or to all the sides for up to 15 micron, or before all the cells are expanding, if some other cells actually come before 15 micron, they will stop there and draw a boundary, and that is how they make a cell to cell boundary prediction using computational based segmentation. This is one example, and here cells are colored, pseudo colored based on the cluster they are classified, and the gene expression is also shown in this case. Okay, so this is some of the way you can do this. You can also do spatial epigenetics. So, it is not just like you can study expression, but you can also look at chromatin accessibility or protein DNA interactions, like cut and tag you can do on this, or like chip experiments you can do. And this platform is called as spatial epigenetics platform, called as Atlaxomics. So, what they are doing here, you put your tissue and then flow barcode. First you flow one set of barcode in this direction, and then you turn your slide and flow the second direction 90-degree perpendicular to it. And doing that flowing of barcode in 2 different directions, what it gives you is a combination of barcodes over the tissue, and then you go and do your experiment on that. So, essentially you can do spatial ATAC-seq and cut and tag. How many people know what is cut and tag? Anybody know? Okay. How many of you know what is chip-seq? So, chip-seq is to study histone modifications, DNA protein interactions and all. Cut and tag is actually chip-seq, but it actually uses a tagmentation based instead of sonication, or sharing is done through tagmentation. So, that makes it easier, and you can do with a smaller number of cells. Okay. So, So you can do spatial ataxic and spatial cut and tag on this, and this where you are essentially, I just mentioned you, like you will flow barcode for one end to other end, and then you flow for the other direction, and this will generate a combinatorial barcoding, and then you use that for you make library out of it and just sequence it. So this is how it looks like. So I think I will just stop at this this point. So I just want to add one more quick thing, and then just I will stop. So one of the main, how many cells you need to analyze for a single cell, and how you actually decide that. Because you will ask people like, this is a question that I always get, like people when they are asking for some kind of a consultation, they will say, hey, I want how many cells I should be assaying. So the number of cells that you need to assay depends upon the frequency of your cell in your population. Suppose you want to analyze a cell, or you want to see any cell type which is represented as low as 1%. So if this is a number that you are thinking about, that okay, I want anything that is around 1% of the population, I want to see that number of cells. You need at least 50 cells for a statistically relevant cluster. Okay. So most of the programs that make cluster, they can actually make cluster out of 20 cells, but it's always good to have at least 50 cells in a cluster. Now, so if you need a 50 cells in a cluster, and your cell is represented only 1% of time, that means you need at least 5,000 cells for your analysis. Okay. But suppose you are only interested in those cells which are more than 10% representation, that means even if you analyze 500 cells, you will get that number. But if you are looking for 0.01%, like very rare cell type, that means you have to sequence almost 50,000 or 500,000 cells to get that rare population. So it's not like everybody you will see like 10,000 cells. 10,000 cells has no meaning. The number of cells, how many you need to analyze, depends upon how rare the cell population that you want to analyze, and this is how you calculate that number. Okay. So I think we have covered extensively. We have looked at almost everything, and just one other aspect. Nowadays, sequencing technologies and sequencers are moving away from. You can close it. We actually moving away from just from the sequencing application. So if you think about sequencer is nothing but a high speed camera. Okay, it's a microscope with high speed camera. So now what they are doing, they are using flow cell to image cell in the sequencer, and the AVT sequencer I have showed you from the element, it can actually do cell morphology analysis, in situ RNA sequencing, then protein analysis on the flow cell. So you put your cells on the flow cell itself, and it will image from there, because these are camera, high speed camera with a microscope. So the flow cells and the sequencers are actually opening up new applications, such as multiomics analysis on the flow cells without sequencing, imaging based applications. So the things are changing, new applications are being developed, new capabilities are unlocked on these flow cells, and sequencers are actually moving from just a element of sequencing, but rather than they are actually becoming more and more instrument to study multiomics, like cell morphology, protein abundance, in situ sequencing, perturbation analysis on scale. So things are actually changing, and this is the most exciting time for genomics. Okay. So with that, I think I'm done with my next-gen sequencing classes, and I'm here for next 2 days. So any questions, feel free to ask. I'm happy to.
So we are saying single cell sequencing with PARSE and 10x as well. So the major problem we are facing that the fixation of the cells. So in PARSE kit, they told us we are usually trying to fix the cells, and after fixation the viability is very less. In the 10x kit, they told us the viability should be more than 80%. So
how fixed cells will not be viable? How fixed cells will be viable?
So in 10x case, the cells is viable.
No, the moment you fix a cell should die. So usually the fixation protocol, they have like something called as methanol fixation. That is what they use. The moment you have fixed cells, you don't need that viability numbers need not be there. Before fixing, the cell viability needs to be 80% or 90%. Okay, because after fixing, cells are dead. You cannot actually
So that means integrity they maintain,
Cell integrity is maintained. The cell membranes are that is what they are looking for. But then, if the moment you fix anything, that means they are dead. They cannot be.
Plus at 10x, the fixation, the biology is maintained. So we have already.
It cannot. How? The whole idea of fixation itself is to kill the cell.
I had a question about like heterogeneity. So like you mentioned that like when we are like extract right like basically like
It end for everybody? No? Okay, what an Eimer.
Will you be able to know how many people are running now? Oh, not really.
At least those. I'm going to try to finish at least the PCA part before lunch, and then if it's still running, it still runs. At least it finished for me, which is the important thing. So we were at that state where we transform our counts. We apply one transformation or the other. I don't know which one did you guys apply. So basically, we got a new object with our count data, but it's been transformed. So again, we have samples in the columns, genes in the rows, but this is the transformed data now. I'm going to insist this transformed data is not used for analysis. We already done the analysis when those two lines, when we call these seq2. So these data, we're going to use it for plotting, like PCAs, heat maps, and things like that. So the next step is principal component analysis, and is when we're going to use the transformed data. And I was saying that we can use the principal component analysis function that DSeq2 provides. And basically it takes the new object that we created, and also it's going to request, if you want to, how many genes do you want to use for this. It's going to be sorted based on the variability of the genes. The highest variable genes are going to be in top, and you can take, for example, the top 5,000 genes if you want to. The idea is the most variable genes are going to be the ones with the most information. Probably it's not going to change if I say 10,000, because I'm already reaching the most variable genes, the one that has more information. You can try. We can change numbers to see how much the PCA changes.
I think it was slow because people have executed the command multiple times.
Could be. I'm not sure. Well, if it's multiple times, it's going to go back and restart. It's going to go back and restart. And it's going to be a restart.
So when I'm checking my kernel status, it is showing as busy, and it is showing executed 8 out of 17 cells.
It says, sorry, 8?
8 upon 17 cells. What does that mean?
Oh, no, that could be the total steps that we have, the total cells that you need to execute, and you have executed 8. No? I don't think so. Or maybe, I don't know. I'm not sure.
You may say 4 out of 6 cells are executed.
Oof. You cannot read it, but I need to do this to be able to show the plot, right? So you're going to get a plot similar to this. Do we have a good clustering based on our condition? It seems so, right? We PC1. So if you could, you can do a plot only using one principal component, right? I mean, it will look like a line and dots on top of a line. But at the end, I mean, PC2 is not telling us much about the story here. It's just PC1 who is splitting normal on the left and sleep deprived on the right. But if you could, you do one PC plot, and all dots would accumulate in one side and the others in the other side. So yes, we have good grouping. The ones in the back or the ones in the front row probably don't see it, but you have it in front of the screen. You also can see how much of the variance PC1 is explaining and how much of the variance PC2 is explaining. So PC1 is having a good separation, but only explaining 37% of the variability in the data. So we could say that, we can phrase it differently, right? We could say that our condition, being sleep deprived, is only explaining 37% of the data, right? Or the variability in the data. So we have 67.3% that is explained by other reasons. It could be technical. It could be, again, we also have a knockout. It could be the knockout, right? We keep track of the mice that were knocked out. It could be that knockout is explaining some of the variability of the data. And again, hidden factors that we are not aware of.
If we change the condition, like we have changed it for the genotype, so we are getting a PC plot with a scattered plot. Right now we have done the comparison between the condition.
Yeah, again, we didn't, here we are not doing any comparison. So we are grouping the samples or coloring the samples based on the metadata. That's it. So we provide exactly the same data. In this case, the next cell. What we're doing in the next cell is basically the PRCOM is the principal component function, the native R function, the one that is included in the basic R, to do principal components. So here, I'm just repeating the previous step using the native function of R. Basically, it returns a new object, and instead of keeping the whole object, what I'm doing is just getting the data points for creating the PCI plot. I store it in here. I'm giving names for the rows, which are our samples, giving names for our columns. So what we're getting here at the end is a new matrix of all the principal components. The coordinates, sorry. The coordinates to be able to plot a new PCA. Oh, and it's going to take forever? No. Okay, so basically what I'm doing here is that obtain all our PCs and our all samples and the coordinates. So now I can take this and use any plotting system and replotting the PCA. why I'm doing all this transformation is because how the data is returning as an object. But don't worry about it. I mean, I'm just showing, you can just plot the PCA using the normal function. I like to retrieve this data because it gives me, or calculate it using the base function, because I'm able to plot more things easily or modify the plots as I want. And we will see how we're going to do it next. For now, the only thing you need to know is we have this GIAN matrix or data frame with the coordinates to plot PC1, 2, 3, 4, and 5, and for every sample. What is PC? PC is the principal component number 1. PC is principal component number 2. When you do a PCA analysis, I explained that yesterday, what you're doing is reducing the dimensions of your data. Your data has, I think in this case, we reduce it to 15,000 genes. So you have 15, no, in this case, we're using all, oh, by the way, in here, I'm not selecting genes. I'm using the whole data set. In the previous step, I chose the top 5,000 genes to the PCA. Here, I'm choosing everything. So it could be slight differences between this and the previous PCA. So a principal component, so go ahead. So the PCA, what you're doing is reducing the dimensions of your data to a new axis or new dimensions, which are completely independent of each other. So this is a new dimension. A PC, a principal component, is a set of genes with different weights. And that, we need to do statistics to explain how all the steps go. But this is a new dimension of your data. You've been reducing to these new dimensions. And each principal component explains a proportion of your data, of the variability in your data. That's what a PC is. So in the previous plot, we're explaining, we're plotting PC1 and PC2. But there are other principal components. I don't even know how to speak now. So another thing that I can do, you remember we talked about the percentage of variability the data explains. When we created, or when we use the PRCOM function and it created a PCA object, it stores everything, the principal components and the importance of each one of them. So to be able to access that, I need to use all these complicated things. It's not that complicated. Basically, I'm access the summary of that object, and there is, let's say, one column or one part of that object that contains all the importance, in this case they call it, or how much variability of the data, each principal component, 1, 2, 3, 4, and so on, it explains. And I'm storing them in a variable. So each variable here is just containing a percentage. That's it. Something that we haven't taught here is how to plot with ggplot. So you don't know all these functions, right? So don't worry about it. But ggplot is a library for plotting. It's really powerful, better than the basic R, and it allows you to do really cool plots. And oh, well, we did one, right? I think the first day. Yeah, we used it the first day.
Francis, there's other thing. So the second Word file that we did not open yesterday is about ggplot, and it has like all the detailed instructions, like how you plot a ggplot. Just run the code, you will actually make sense of it.
So basically, I'm giving our metrics. I'm asking plot PC1 and plot PC2, and then modify that plot with other things, right? Let's annotate every sample, give the label for x-axis and y-axis, and use the percentage of variability explained, add it also to those labels, right? And they come with different themes, so you can play with the colors and all that. So if I do it, I store the plot on a new variable, and then I print it. Or I show it on the screen. Sorry, I missed one step, right? Sorry, I didn't run this. It's definitely nicer than the previous one. So now we have all the labels, all the samples being labeled, and pretty much it's telling us the same story, right? That it's separating normal and sleep deprived. And we also printed the percentage of variability explained for each principal component. It is slightly different, right? I think it was 37 in the first one. Now we got 34, still in the same range. So clearly, our condition of the study is not explaining everything. But enough at least to separate. Well, okay, one thing we can say for sure is the main source of variability in our data, right? Is it's been sleep deprived or not. That we know. Okay. Can you plot PC3 and 4? Let's plot it. Actually, this command, this ggplot could be shorter. Now that I realize. Any problems plotting PC3 and 4? Everything good? I shortened the code, so if it looks different from the one that you did it, is because actually there were extra information that is not needed for ggplot to plot it. Thank you. I guess you could.
Yes, please. Actually, my end is not working, but we have made PC3 versus PC4.
Okay, good. Yep.
No, we have plotted for PC3, PC4. So the variability is 9.5 versus 6.5.
90? No.
We have plotted for PC3 versus PC4.
It cannot be 90.
It is 9.5.
Oh, 9? Yes. 90? No. Okay. Yeah.
नहीं वो तो PC1 versus PC2.
Oh, sorry. I did a mistake.
It
My
was
bad.
almost similar, so we have to place here.
My bad.
9.5.
Yeah, yep. I made a mistake. Thank you. I think Bonnie already showed you how to do a bar plot. So if we want to access of the percentage of how much the percentage of the variability is explained by every PC, we can access through the same way we did before with summary, PCA, importance. This is a section that is proportion of variance. Here I'm just multiplying by 100, so we get a percentage instead of a fraction. That's the only thing. Yes, I mean, by default, if you do principal component analysis, they are always sorted. So, and the first two are the ones that explain the most.
Where the two, acha, it is even odd number which is coming in. Who is not here, code is not here.
Oh, that's just the labels. But the second column, it's two.
Acha, there, it's there.
It's there, yes.
Column.
Well, before that, I mean, PC3, PC4 is not telling us much because we don't have much information. The only we have is we know that it's a sleep deprived or not, nothing else. So it's difficult to come out with something here. There's no specific separation of anything.
So this PCA plot will always be on one end.
Sorry, what?
PCA plot will always be on one end.
Oh, well, it's the first one you're going to look at it. But you can look at more PCs if you want to. I don't have a good example, real data, why you can go 3 and 4, but the example that I did yesterday, the separation by treatment was PC3. So if you don't plot it, you're not going to see that separation. So, but what we know, right, is PC1 is separating a sleep deprived and not. So we know that the main source of variability is that, is being a sleep deprived or not. The other sources, we don't know.
Distribution of genes is distributed over the dimensions. So it depends upon your data. Again,
Variation in the data.
yes.
I mean, I'm trying to explain what the PCA tell us. That's it. It's me putting an explanation on it. The principal component analysis doesn't know anything, right? It just knows every sample, and it's doing a principal component analysis based on the variability in your samples. That's it.
So that's what I'm talking.
Yep.
So we have variability in our samples. So
Yep.
we are getting the data between the samples. So
Yep.
PCA plots is just the description of the variation in our data.
Correct. In a way that you can read it,
Yes.
going from 20,000 to only 2 dimensions.
Yes.
Yeah.
But my point is, so it is distributed upon PC1 versus PC2, PC3 versus PC4.
Yes, because they're totally, that part of the principal component analysis tells you that every principal component is completely independent. It's orthogonally independent from the next one. That's why you have it completely divided.
No, dependent.
There's no overlap.
Independent.
It's a dependent, sorry, independent. I meant that.
No, suppose you have 20,000 genes. So my understanding, what I
Yep.
understand about PCA plot is, suppose you have 20,000 genes in your data.
Yes.
Yeah, for 10 samples,
Yep.
distributed over the 10, 5 in the treated or
Yes.
non-treated groups. So we have 2 groups, right?
Yes, but the groups doesn't play any role in principal component analysis. Yeah, okay. To be clear.
I'm giving the example.
Yeah.
So we have 2 groups, treated and treated,
Yeah.
and we have 20,000 genes.
Yes.
The variation is quite much in those samples
Yes.
or groups. So PC1 versus PC2 is is the data of approximately 16,000 genes, and PC3 versus PC4 has data of 2,000 genes.
No. They can have all of them. So principal component can be all genes, but they are weighted differently.
Why? So why they?
Well, then we can go into statistics, which I'm not an expert, right? But that's the main idea. It's a linear combination of all the genes, and every gene is weighted differently. So genes that don't variate, they have a weight of 0, meaning they don't contribute to the principal components. Genes that have high variability, again, I'm not an expert. I remember doing it by hand when I was an undergrad, right? I remember doing principal components by hand just to learn how is it, but I don't even remember the details. So I'm not going to be able to explain you the details.
Variation in the gene expression. Suppose PC1 is telling us 50% the variation or 60% the variation spread over the PC1. PC1 plot is telling me the maximum percentage of the variation in the data, not in the sample.
So should we stop here? Yeah, okay. Any questions about principal components? How we plot them? I gave you 2 options. If you like ggplot and you want to learn, use Bonnie's notebook to practice, or ChatGPT. Don't use my example because I also make mistakes, right? So I know this section here, you can skip it totally. You don't need it. If I tell you the truth, I use ChatGPT a lot for ggplots because I'm not able to remember all the parameters. I don't remember them. Okay, let's break then and be back at 2, the latest. anyone remembers what loadings means in a PCA? We mentioned really quickly, but maybe somebody remembers. Loadings, weights, they call it. So again, the PCA object contains a lot of information. We access the variance explained by every PC, the actual matrix of the data, but also contains the loadings. So here I'm accessing the loadings for PC1. The loadings are the contribution of every gene for that principal component. So if we want to know the main genes creating the separation between normal and sleep-deprived, that's the PC1, right? That's the one that's separating our condition. And we want to know, okay, what are the main genes contributing to that separation? I'm going to look at the loadings. So this long thing here is just accessing, sorting, and giving you the names of the genes. That's what it's doing, right? And I'm using the pipe. So I'm storing the loadings of PC1. I'm getting an absolute value. I'm creating the sorting, transforming to a data frame, adding the gene, and then I don't know I forgot what I'm doing here. But basically, what I'm trying to print out is the loading. So I want to know what genes are the ones that are contributing the most to our separation. And here you have them. And the loadings for each one of those genes. And the list, of course, continue. You have even more. I'm just printing the top 6. We don't do these often, but you could. You could explore your data in that way and figure out, okay, what genes are contributing for PC1, the ones that contribute the most. So those are the top genes.
So DBL here is the log 2 fold change, the last column.
No. It's the loadings. So there's no, again, our analysis has been done. Now we are playing just with, remember, we transform our data. And then we did the PCA. PCA is just you saying their own analysis. There's no related to fold changes. There's no p-values, nothing. This is just the loadings of every gene. So this is actually PC1. Is a linear combination of every gene with a loading associated to every gene. So what I'm doing is trying to figure out what are the main genes contributing to the separation of PC1 that you saw in PC1. So these are the top genes. Again, it's not something we do commonly. I'm saying that it's doable. That's the only thing I'm showing.
Can we get the number? This is just the top 6, right? Can we get the number of the genes?
It's going to be all of them, probably. I don't think they filter for anything. You're going to have loadings of 0. If we go to the tail, right? I'm assuming R has tail. Oh, sorry. If I do... No, it doesn't need to be positive, negative expression. It's just doing a PCA. There's no expression here. So all genes are present, but with a tiny, tiny contribution. So, okay, Bonnie was asking yesterday. Okay, Bonnie, pay attention. How we can save plots. So there are different ways you can do that. So if we use ggplots, they have their own function that you can use to save plots. We can also use this own function that we can call a. So the way that I prefer to save plots is using PDF. I don't know if you ever worked with Illustrator with vectors, vector graphics. So vector graphics or raster images. So raster images are just pixels. You cannot modify them except if you do something special about it, right? So if you use vectors, so vectors, what it contains is information about the lines, the fonts, the text, and everything. So PDF allows you to save as a vector. So if I want to save a plot as a vector, later on I can modify it. I can open in a vector graphics editor, and I can change the font, I can type some text, and so on. If you have a raster image which has pixels, it's more complicated. I'm not going to be able to change the font. I'm going to have to put something on top of that font or do some manipulations that you don't need to if you can save the vector graphics. So I generally recommend to save it as PDF, but you can save it as a PNG, JPEG, or whatever you want to use. So the way it works is you open a device, they call it. So I give the dimensions. I think this is inches. So this is just a piece of paper size. And then I plot whatever I want to plot. I keep plotting, 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 and then I close the device. And everything between these two lines, whatever I plot, is saved in a document, which has this title. Right? This is a way to do it. You could do it that way.
So if I will save it in the PDF format, then I will not able to do any changes over it, right?
Yes, you could. Yeah. So that's what I'm trying to tell you. So PDF is going to store it as a vector graphic.
Okay.
So it's called scalable vector graphic. So you're going to be able to actually modify it. If you open this in Illustrator, you're going to be able to modify the lines, change the thickness, change the text, whatever you want to do with it. Illustrator you need to pay. So Inkscape, I don't know if anybody works in Unix, probably you install Inkscape. I think it's called Inkscape. Is Inkscape the one that the vector graphic, I think? Well, it's the one that you can probably use to modify these for free. I'm assuming Inkscape exists for Windows too. So we saw this plot earlier, which is the bar plot of the importance of each, or the variable explained by every PC. So if I run this, it doesn't give me any output because I save it directly to a file that it's here. So if I double click, here is the image that I just saved. So I'm asking you to save PC1 and PC2 in a single document. Give it any name. You can call it pca.pdf and save it as a PDF. Dimensions, you can give it or not. It's up to you. I think it's, what type of paper you use here? What is the size? A4? A4, okay. I think it's this letter size, the American letter size. So any problem saving the file? Did you create a PDF? Don't replot them. I don't know if you remember that we store every PC. We created an object that contained the plots. You don't have to replot them. If you go back, right? We store PC3 and PC4 in here already. So if you run already this, that's enough. You don't have to replot them. I'm assuming it's enough. So if I double click to the image, here we go. So now we have 2 pages, we have 2 different plots. A little bit too stretched, right? We should change the dimensions if we want to have nicer plot.
So there is no way to, like,
So we scroll it up and up.
click a button kind of thing to download the files. I mean,
Yeah. So if you want to download, well, I don't know if we should teach this. So Amazon charges for downloading stuff. But yeah, you can. Again, we're going to share everything. So at some point, we're going to put everything available. PowerPoints, I'm assuming, too, if you want them. I think at the end, nobody looks at them, but the Jupyter notebooks will be available. If you do right-click, and there's a section that says download, so you just can download it.
yeah, don't download because maybe AWS will have.
And you don't need to download it. If you have your own Jupyter notebook in your computers at some point, it's going to be in your hard drive. So you don't need all these operations on the left. Just added for fun this section here, which is plotting 3D-dimensional PCA. So it's possible thanks to the Plotly library. So if you try, you're going to get some 3D dimension here, plot, and you can interact. So R is really powerful too when you can do interactive plots and things like that, but we are not going to explore it. But there's a ways to do 3D plotting in case you needed to plot 3 dimensions for whatever reason. Again, I don't think this is practical necessarily, but it's fun. Okay, so heat maps, that's something we talked also about yesterday. For that, we're going to use a library called Pretty Heat Maps, P-Heat Maps. The question is, what genes we're going to be plotting? For now, we're exploring. We don't know what genes are differentially expressed or not. They are in our object, but we haven't checked that. But what we can do is, for example, in this case, I'm picking the top 200 most valuable genes. Based on the, again, I'm not touching yet the analysis. I'm just using the transform data. Basically, it's calculating the variance for every gene, ordered them, and I'm just picking the top 200s. I'm using this dot to say whatever output is going to be a data frame that I'm taking 1 to 200. That's pretty much what I'm saying. Or vector. Oh, yes, sorry. Oh, that must be correct. Hold on. Is this a data frame? That's a good point. In R, they use index 1. The 0 doesn't exist as an index. So I'm not sure why I said that. Ignore it for now.
In front of that, it means it's disabling.
Oh, yeah. So this is not a line of code now. This is just a comment. So actually, this should be, I don't think it should be a 0. It should be a 1 because in R, it's index 1, not index 0. But this is not going to run anything because it's just a comment. You're correct. So one thing, again, it's always good to explore what we're doing, right? So if, oh no, that's the one I want to do. So anybody knows what is this? So I'm not getting genes. Yes, but these are not gene names, right? This is the index position in our table. It's just where the gene is in our table. That's what it is. No, is the position in the table. It's the row. So the topmost variable gene is in the position 14,400 something. Because I'm going to need this to access the genes on the table, basically by position, not by name. Oh, I'm saying it here. So if you read it, it contains the raw indices of the topmost variable genes. That's what it means. So I'm asking what this section here does, basically. We used these brackets before a few times, and what we're doing here, remember, separated by comma. Whatever's in front of the comma, the first number you place here is the rows. Whatever you place after the comma is the columns. If it's empty, it means everything. I'm giving in the indices of the most variable genes. So you are getting only the rows for the most variable genes. So I'm just selecting our modified counts to get only those for the genes that I care. So those are the ones I'm going to be plotting. And this is just predicate map function telling everything we need to do, right? So I'm telling them cluster by rows, don't cluster by, don't show the row names, cluster by column, and explain the rest in a second. Just, we can plot it now. In this case, compared with the case we talked yesterday, right? We said that heat map from a paper was selecting genes that created the separation. Here we are not selecting genes creating any separation. We're just picking the top 200 most variable genes. So this clustering is already telling us that a good clustering for our sample, just picking the top 200 genes. But this is typical, right? We generally present on papers a heat map with clustering for the samples, clustering for the rows, although again, I don't think this gives us any information. It's really hard to read, and we don't make any comments generally on papers about them. So I'm still not sure why we're still plotting them, right? And generally, just looking at, you already know that is a group of genes here, these clusters here that lower expressed for normal, and this is higher expressed by sleep deprived. So I don't know if anybody knows. Do you know what this part here means? A scalable row. Do you notice here? I'm expressing, or I'm plotting the count values log transform, right? That's what we're plotting. But why we get negative values? Doesn't make any sense. I don't have negative counts, even if you do the log transformation. So it is because we're scaling. Try to plot it without the scale. Say, so if you remove, right? Super hard to read now. I'm getting a completely different story here. And if you have a gene that is really highly expressed, it's going to be red, and everything else is going to be blue. So what we do with the scaling, oh, sorry, not this. Whereas we are, sorry, normalizing or z-scoring. You know the z-scores? So you're z-scoring the whole row in that sense. So these are z-scores actually, right? So you get the average of the row, right? So the average of the row is going to be 0, and the standard deviation is going to be 1 or minus 1, right? So this is what you're doing. So basically what you're telling here, I'm doing a really poor job explaining this. How is called the z-scoring system? Is normalizing? So you're scaling the row to have an average of 0 and a standard deviation of 1. That's what you're doing. So basically, 0 is the average of this row in expression. That way you can compare every row. If you use absolute values and you have one gene that scales 100,000 and the rest are values of tens, and you try to plot it, it's going to look like around red and everything is going to be blue. In a way of scaling, what you're doing, you're going to be able to compare the rows in that way. Standardizing the row. That's the word, right? So you get a z-score.
What are the bars above the blue and the red line? What are those?
Oh, so these clades? Yeah, so this is a clustering. This uses a method of distance clustering, like a phylogenetic tree. So basically, no, you're calculating distance between samples in here and distance between genes here. Yeah, but it's related to the idea of phylogenetic trees, right? So you calculate the distance based on the expression, and you beat a cladogram on top to know how close to each other they are. So basically, you have two big clades, and they represent the normal ones and the sleep deprived. And because we picked the 200 most valuable genes, we already know that that means that we have a good clustering.
And why is it so crowded on the y-axis?
On the left, because we picked 200 genes. Let's pick less, right? Yeah, so, my bad. I think that probably you already knew, but maybe I didn't explain it well. Every row is a gene. Every column is a sample. I mean, you have the names of the samples here at the bottom. We don't print the names of the genes because they wouldn't be readable. I mean, it's already hard to read this. Imagine having 200 gene names here. I mean, I think the exercise that comes later makes you plot something else. Yeah, plot the top 20 and show the gene names. So because 20 genes are going to be readable. Good. Yeah, well, gene names is using too much space, right? Yeah.
Reduce the font size.
Yeah, we could, or let's not show it.
You remove the column names, there's like enough space.
सर फॉन्ट साइज चेंज करने से वो जीन्स यहाँ पे दिख रही है
These are not the only options within pretty heat maps. You have a lot of different options. Actually, there is one that is file name. So if you give it a file name, you would save it as that file name. So what happened now with the clustering? Right? We don't get that nice separation. So if we do a PCA with only 20 genes, similar situation is going to happen. We're not going to have good separation of the samples. So the 20 top genes doesn't explain the whole story either, right? And don't run this section. I think we can skip it. We don't have to run this, just in case it takes too long. Does anything that would take long? I don't know, I'm worried that accessing this would take a little time, but let's see. Don't run it. Let me try first. Oh no, it's fast. You can do it. Just in case. Should I move on? Still people trying to plot the heat maps? We'll have more chances to plot more heat maps down the notebook. I mean, while we wait, spend some time here trying to understand what we're doing. Everything good in this side? Seems so. Okay. I mean,
This is the data of normalized mean.
yeah, we're asking DDS, give us the counts, right? And we ask them how, normalize, row or not. So probably if you say false here, you're going to get the row counts. But we want the normalize, right? And this goes here, right? So basically we're asking, give us a data frame. That's the structure of R for tables, right? And we store it in normalized counts. What about here? What are we doing the second line? Any guess for the second line?
We are defining the row names, rows.
We are getting the row names, which are this, right? And what are we doing here? We're creating a new column. Remember this is the symbol of column to access columns. If this doesn't exist, it's going to create it. And we're assigning the row names to a column. It's the name that I give it to the column. Did I keep it? Did I do something with it? No. I created a column, so if you, well, we don't have to do it. I created a column called gene. You can give it any name you want. Just it's arbitrary gene name. I mean, this is also pretty readable, right? Yeah. So basically, we are separating the column we just created in two new ones, called ensemble ID and gene symbol, by this pipe. I know this forward or backward slash, I always get confused how to call them. They are necessary because the pipe could mean something else. So just to be sure, we're telling this is the pipe symbol, basically by this pipe. We're splitting this into two new columns, one called ensemble ID, the other one symbol. And then, so we have this table, and we rewrite the table with these new two columns. This is what we told you before. There's that library called OpenXLSX, so XLSX. And what we're doing is just storing the count data in this file and giving a name to that specific sheet.
So this is for Excel.
Yeah, if you want to create Excels in R, this is a way to do it. So I'm storing a data frame inside of here. That's the short story about it. Oh, finally, we've arrived to differential expression results. Okay, the way to access the results is just calling results into our object, to the DS object. We have an example here. So this is the third step. So if you want to summarize, and I think I've done it at the very end, we created one object. We give to the DDSig2 our data with the count data, metadata, and our formula, right? Then we asked DDSig2 to do the analysis. Actually, our third step would be access the results. With these three lines, we've done the analysis. Everything we've done between the beginning and here, it was just exporting the data. DDSig, I think it's the vignette recommend. I don't remember what I read it. So you can actually specify the alpha you're going to use for filtering. They recommend that because it's used internally for filtering genes that are not important, right? What we said, there's an independent filtering process that I'm explaining here. And I don't know if we need to go into many details, but internally, DDSig2 makes a decision about genes that are very low expressed. I'm not doing anything in your analysis. And to some extent, it's important to filter them. And well, if you read, you're going to know why, just because of this. So the more tests you do, the more adjustments you're going to have to do to your p-value, right? So when you do analysis or a statistical analysis, right, you generally say, oh, I'm doing this t-test, right? And if it's below 0.05, I'm calling it significant. We are doing 15,000 tests. Let's assume we don't filter anything. We have 15,000 genes. We're going to do a test for every gene. So just by random chance or by definition of the 0.05 false discovery rate, what we're doing is assuming that 5% of the time, we're going to have a false call. So 5% of 20,000 genes, how much is that? 1,000, 2,000? No, 2,000 would be 10%, right? So 1,000 genes are going to be called we're going to call it significant when they are not significant. That is why we apply correction. When generally we do a lot of tests, what we do is to apply some sort of correction, right? We call it false discovery rate, Q values, p-adjusted values, depending on the technique we use. So if we remove those genes that are useless because they don't have any, the count is too low, they're zero, we don't have to do a test. So the correction is going to be lower. So one of the reasons why we do the independent filtering is called is that. So we want to correct for that. In any way, they're going to have a very low count and they're not going to do anything in our analysis and we don't care about them. And if you really care about how it's happening, the independent filtering, there are some options that you can tweak if you want to. And so if you read the manual, I think it tells you how to do it. I generally leave the default. I don't think you need to mess much about that. So
Sometimes it comes true and false in the data, or the false discovery rate is less than 0.01. So,
so if you want to be more stringent about your false discovery, about your alpha, you can reduce it when you do this. You can change it here,
to 0.1.
for example, if you want to.
Otherwise, the 0.5 is sufficient.
Yep.
For the
I'm assuming, I don't know if there's a default in this value, maybe the default is 0.05, but there's nothing bad or being explicit about what you do, right? So changing the 0.05 changes the way that independent filter works.
ideally, we should remove these genes if it is false discovery rate is less than 0.05.
No, you don't have to remove that.
Okay.
So if
we are doing data manually, like here we are giving command. So if that we have data and it's showing false discovery rate of less than 0.05.
no, that was when it would be significant, right? That's your p-value, your adjusted value. No, this alpha is the cutoff that you use to say if it's significant or not.
Okay.
Right? In statistics, we call it alpha, right? The p-value, alpha, p-value. So exactly, it's the p-value. I don't know why. Yeah, it's better to call it p-value. So this is going to be the cutoff you're going to be using. So if you know you're going to be using 0.05, it's good that you do it, or you indicate it when you access the results. So again, the only thing we're doing here is reading the results from this table. We are not recalculating anything. Again, I'm using head here to obtain the top of the table. The results table on DD6 already tells you what you are comparing. It tells you your condition as lead deprived versus normal. It uses the wall test. Sorry, I didn't give many details about the test, what is used, but that is part of the type of analysis we're doing. This is not a t-test or anything like that. We're using wall tests because we're assuming it's a binomial, negative binomial distribution. That's how it's normally the gene expression data is distributed. We have a bunch of columns. I'm assuming a lot of you are familiar with them, right? The fold change. I don't have to explain much what it is. Log2 fold change is a log2 fold scale. This is a log2 fold change, a standard error. This is a statistic of the wall test. The p-value, and this is the p-adjusted value we were talking about, right? So if you do multiple testing, you need to correct for multiple testing. So this is the p-adjusted value, and it uses the B and H, it's called, no? The Benjamin-Hort correction. So this is how we corrected this p-value. So now you have the results. So you could do any filtering with them. You can download them, have it in Excel. If you do summary results, you're going to obtain something like this, and already it's telling you out of this number of genes, these are the ones that are significant with positive values, with a positive fold change. These are the ones that are not significant with a negative log2 fold change.
But then together make up around 40%. What happens to the rest? Like there are 8%.
So the 60% or the 59%, it's not significant. They are non-significant. So these are only the ones significant with a p-value, adjusted p-value below 0.05.
So those non-significant ones are not coming to the list.
No, they are in your list. So they are in your table. They are here. So for example, this one, this is 0.95. Or this one, this is 13. These are non-significant. This is just a summary of the table on top.
But then can you put a command and tell that let us see that rotate slowly only the...
Oh yes, you can filter it. This is R. Once you have a table, you can apply any filter you want. Okay, so there's one more thing. You can still tweak a little bit your data. Are you familiar with the MA plot? So the MA plot, I think it comes, I remember used them already with microarray data. I don't remember what MA stands, but I think it's for the old times. They kept the name. So basically here what we're doing is, so the mean of normalized counts on the x-axis and the fold change here. So basically these are the highly expressed genes and the lower expressed genes. And you can see in the fold change, there's a lot of variability, right, compared with the higher expressed genes. The blue dots are the ones that are significant. Yep. So if we want to plot fold changes, we want to sort by fold changes, we're going to be biased by how much variability are here in low expressed genes. It's really hard to tell if you have a count of 1 and a count of 10. Yes, so the calculation may tell you it's 2 times more expressed. Is it? You don't have much information. Really, you have that much information to be precise how many times it is? It's different if you're working 100 levels, right? You're going to be more precise about the fold change in here than in lower counts. So there's a way to correct for that if you want. This is not only going to be important for plotting fold changes or if you need to sort them by fold change. So there is a process. Well, I'm already answering the question, sorry. What do you notice in the MA plot? So that the variance of fold changes is higher in lower counts than it is in higher counts. So there is a function in DSIC2 that is called log fold change shrink. That function, what it does is to reduce the effect of the lower counts to make the fold changes for lower count genes smaller. So basically, you provide the object and your result table. I think it's better if you do this. And there are different types of shrinkage. So in this case, we're going to use this one. But I mean, there are others. You can read about them.
So it will remove all the low count genes with having higher.
No, we don't remove anything.
No, it's not removed.
No, we don't. At this point, we're not removing anything else. We just have a table stored in this variable called res. Right? If you go up, Yes. Yeah, okay, it works.
Say I will change battery is low, sir.
I don't know if it's low. It's working. If you're going
No, sir. Red light is blinking now.
Oh. Okay. Take your time. I don't like the mic. Okay, save it. I mean,
It didn't happen.
what do you mean it didn't happen?
One more row is the same. One more column.
I didn't touch anything. I mean, I ran my code. You have my code. It should be the same.
No, it is not coming input table.
Gene ID and sample ID, they didn't separate in the next table, the neat one. He said one more key column has been created.
Yes.
Okay, okay. At the end, okay.
Okay,
Yeah.
okay.
So save it to a file. So now you want that table in your computer. Maybe you want to use Excel for filtering or for whatever reason. So you can save it to an Excel file. Anybody has questions? Am I going too fast? Too slow? Stop me, ask questions, everything I'm good to do.
Just right pace.
Just right pace? Okay. And again, if you can always read everything I'm saying, I'm pretty sure is there. I don't think I'm not saying anything new or not present there.
Can this all material be downloaded? Like what you have put in the
Say it again, please. I didn't hear you.
like all the material of all the days can be downloaded somewhere? Like,
Everything we're doing, so I don't think it's worth to download these results we're having now, but the Jupyter Notebook will share it for sure, yes. We're placing some disk space, you can download it or send it by email. We figured out a way to share it. Like Google Drive or OneDrive, we create something like that to share everything, yes. There is an example a little bit above this. We ran already one example. We saved the normalized counts in an Excel before. Oh, and to give another example how you can save it, because it's, how we call, fine with uppercase, right? Don't copy me, do it yourself. All right, I'm gonna call it like this. No, don't copy me. No, I'm gonna wait. So I'm not. If I ask something, the solution is already there. I'm not asking anything new. Here you have your answer. We've done it before. And I think the next time if I use this Jupyter Notebook for something, I'm going to correct everything so we use pipes all the time because I think it's more readable. But okay, good. I mean, something like this should work. I think we give it more options in the other one, right? What do we give? Oh, we give the sheet name if you want to, sure. Sheet name was like this. Don't copy me. Why people copy what I do? I mean, it's as simple as this. You don't have to give more options, but you could. I think there is an option that you can give, one argument that allows you to freeze the first line, the headers. I think it's really practical, but I don't remember how it's called. It's called freeze. It's freeze. Freeze it. You need to indicate what row you. I mean, I don't know. I'm gonna try this. Oh, it works, but I don't know what it does. Let's see. Don't download anything, okay? I'm just showing the results for you, but you don't have to download anything. We are not going to use the Excel for anything. No, it didn't freeze anything, right? No. That option didn't work. I don't remember how it's called. ChatGPT it and see if you find it. So you don't have to download anything. Don't download it. I'm just downloading to show it to you. So if you save it as an XLSX, you can open in Excel. You can apply filters however you want. If you like Excel to work in Excel, this is an option. You can do that. But you know so much R now that you don't need to do this. Oh, what I'm doing with that. Okay, next step. We're going to improve the volcano plot that Bonnie did yesterday. using the enhanced volcano. Well, we improve it to some extent. So a little bit flat, but we can, if you change the options, you can make it nicer, just extending it vertically. Well, we can make it nicer, but so we change the options in the display. So we can increase this. Oh wow! But I made okay. Let me way too big. Sorry, I need to make that. Sorry for the people in the back. You can change the options for enhanced volcano. You can make it as wide as you want, as narrow as you want. There's a lot of options you can play with it. I don't know if you remember this gene. This is the one that was the top one in the loading for PC1. That was the one over there. Probably those ones are over there here too. So do you know how to read the volcano plot? I don't know if Bonnie mentioned it. So basically what we're doing here is plotting every gene, the fold change of every gene. Right? The ones on the right side are positive fold changes. You have the 0 here. The ones here are the negative fold changes. And here you have the minus log 10 of the p-adjusted values. And we are creating some cutoffs, right, with the vertical lines. This is the log 2 fold change cutoff. And then we are coloring depending on if they are in the mid section or the right section of those. So these are going to be the top most differentially expressed genes, and we have them on the positive side. It seems that we don't have any reduced expression below log 2 fold change on the left side, right? So these are a good way to summarize the whole table in just one plot. The plin 4, I don't know, it's not necessarily the most variable, but it's the one that has the highest fold change and the lowest p-value. Yeah. What I mentioned about it is that it was the gene with the biggest loading in PC1. So the one that PC1 was the one separating sleep deprived from non-sleep deprived. Okay. So is sleep deprivation affecting the fat storage? It could be.
People who lack sleep, they are totally obese.
Yeah, okay. Yeah, I heard that before. So that I think plot counts is from DSIC2. So this is the gene that we're talking about. You can just plot it with this function here and separate it by our condition. And you can see that is a big difference, right? Not only a big difference, but also really tight expression, right? We're pretty confident about the variability within groups that this is really significant. Okay, let's practice a little bit the piping and selecting genes and filtering. Can you answer these questions? Using filtering. So try to answer the questions using filtering. So for example, store the significant genes in this variable. Store significant genes with a fold change of 1. I said here, yeah, fold change of 1 in this one here. And then use these genes to plot a heat map. So the genes that are here, use them to plot a heat map. Remember, to plot a heat map, we are using the, how we call it, RLD data. It's not our original data, it's the log normalized. Sorry, R log transform, not normalized. I'm already giving you half of the pretty-hat function, so basically you need to indicate what we are plotting here, right? We're missing an argument, the main argument. We've done some of these operation of selecting. What else we use? I don't know if we use other ones. I don't know if we need anything more than selecting. Oh, filtering. Filter and select may be enough to do all this. I'm not getting away with it, damn it. So if I do this, in the volcano plot, previous volcano plot we had, I'm going to change the title. Right? So all the red dots are 111. So my question was how many red dots we have. Not only the ones on the right side, I want both sides. Do you need to change the colors for the downregulated ones? If you want to change them, you could. I don't think you need to. I mean, you have the legend on top, the legend, the only thing. Right. The legend tells you blue are the ones significant, red are the ones that are significant and are above the threshold. That's what it means. So how is the news heat map? No? Okay, good job. Exactly. I mean, this you haven't even. Oops. So again, I'm not saying anything new. We already plot a heat map, right? What is it? The easiest thing. I'm giving you tricks how to answer the question. So we need. This is the information we need, right? But instead of the top variable genes, we need to provide. So you can even come here, copy this. Come on, you can do it. You should have something similar to this. You have less genes, right? I noticed. Oh, okay. Oh, you didn't.
What is gene
You just created that a moment ago. What it is? What is genes FC1? Is it this? After filtering and getting the row names. So these are the row names of the genes that have a p-value significant and an absolute fold change bigger than 1. That's what it is. So this is our transformed data, and I'm asking getting the rows for these genes. I don't care about the rest. And do a pretty heat map. That's what it is. So that's what I was saying about that paper. That paper showed 10 genes. And if you do the clustering, right, you're selecting genes that create differences. So it's expected that you're going to get a good clustering because we are selecting the genes that are different, and you get a beautiful separation, right? And you have really blue section, really red sections. You're selecting genes that are already creating that difference. Okay, summary, right? To do an analysis in this seq2, or sorry, anyone have questions? Period heat map questions? No? Should I move on? I'm going to wait. Okay, I wait.
So we have to annotate, right?
You have to annotate?
In the heat map. So our heat map is showing error and the argument mat is missing.
Yes, that's what I've been saying from the beginning. You are missing an argument here. That was the only thing you needed to add to plot it.
Yes.
You need to provide the matrix, right? So I'm going back again.
So we have to SA and the fold change
So
value. Yes.
no, not the fold change value. So here, What is in here?
The fold change part.
No, no, no.
Genes with fold change part, no?
Yes. The gene names, the raw names of the genes that have a fold change bigger than 1 and are significant. So I'm selecting, again, the square brackets around a matrix or data frame, right? I'm selecting the rows. I'm giving the names of the rows that I want, and I want all the columns, because I'm not selecting columns after the comma. We already done this before, the pretty heat map. So what I was asking is you needed to fill the gap here. We needed to give the data. Are we good? So a basic analysis, if you don't do any exploration, right? We can summarize it only in 4 lines, or even in 3 lines if you want to. Right? With these 4 lines, you have a full analysis in DSeq2. Without exploring or plotting anything, you could summarize the whole day like this. I'm going to ask you to do the exercise just below. Again, you only need these 4 lines. You don't need anything else. You can do the exercise below. She's waving. I mean, in our metadata, we created this column. We can have call it anywhere, anything, anyone, anything you want. We could have had something like treatment, and still have 4 different levels of treatment. I will insist a little bit more if we have time about how to define the design and the formula. Okay, have you tried to print the top 6 lines of the results? Say, what do you see different here? What is the problem?
It's showing the same, I think, because that was also 1116. Here,
Good point. Good point.
it's showing the same data. So,
Is it showing this? No, it cannot show the same data that I'm thinking of.
so the average is
Well, the base mean is the, because it doesn't matter how you do it, it's going to be always the same.
so here is wild type sleep deprived versus wild type normal. Earlier we had wild type normal.
Earlier, we classify all the samples without considering the genotype between a sleep deprived and normal. Here we split it in 4 groups, right? So here, the main problem now is that we only obtain the results for 2 of them, right? In this case, only wild type. Why? Because we define the levels to be wild type, our baseline, right? And the rest gets less important. But, I mean, you have 2 options. You could relevel everything in a way that you can see the comparison you want, but you don't have to do that. So everybody reached this point? Yes? The background too? Good. Did you see the problem now that we have? That we have 4 levels in our treatment, right? We have wild type as normal, wild type as sleep deprived, knockout, normal, knockout as sleep deprived. And when we access the results, we only get one of the how many possible comparisons? Whatever. If you do pairwise comparisons, depending on if we change sides or not. But we can have a lot of comparisons, right? But we only get one. So let's imagine that we want to access the same comparison between a sleep deprived and normal, but within the knockout. So there's ways to do that. And that's the next step.
So if we use the condition value groups, so it's taking by themselves that wild type sleep deprived, oblique normal there, but we have given just geno condition.
And that's enough for the analysis we want to do. You don't need to give anything.
It's not analyzing all the groups.
Yes, it's analyzing all the groups. So everything you've done so far, it analyzes all. So inside of our results object, not inside of our result object, inside of DDS, you have all the results of pairwise comparisons.
But it's not visible here because we have chosen head for the.
No. Don't go ahead. You have how to access here. The answer is all this paragraph. So the question is, they ask me, do the results contains everything, or the DDS, right? Because we are only obtaining one of the comparisons. So if I go to DDS, so our DDS object, how we access the coefficients. Remember, we can access, I thought it was not coefficients, full name, right? It wasn't coef. Now I don't even remember. Like this? Oh, yeah. Okay. So if I access the coefficients, so our fold changes, everything is here already. Or almost here. I mean, the DDS object doesn't have the p-values, the q-values for all the comparisons you can do, but they are easy to access, right? So we can use the contrast. You can request, you are not going to use condition in this case. This is just a general example. You're going to call the column that you want, and you're going to ask, comparing whatever you want to compare against another thing. And that way you're going to get the results tables that you want. This is, for me, is the easiest way to access. There's a different way to access the results, but I think this is the easiest. You already indicate the column we are analyzing, right? But you're going to have to indicate it again. So this is what we want to access. And you're going to compare, I don't know, let's access what we said, the knockout, the sleep deprived versus normal. So that's what I am asking next, right? I'm asking something like this. So do this. I'm assuming we are missing something here. Oh yes, of course, we're missing you need to fill the gap between these parentheses and fill the gap between this, and then this is complete. Right? This is just defining what is in that column. It's even better if we do this, right? These are the 4 levels that we have in that column. Before we only had sleep deprived and normal. Now the problem is we have 4 levels, so there are a lot of pairwise comparisons that we can do here. So what I'm asking is this. Use contrast, as I explained earlier, to access this comparison and plot an enhanced volcano. You don't have to touch the enhanced volcano, but you're going to have to fill the gap here and here. So we are able to run this without an error and plot the enhanced volcano. And the answer pretty much is just about this. Again, I'm not asking anything new. You just need to apply the same logic, the example that I'm giving above. Did anybody plot the enhanced volcano? Not yet. You don't get tea until you finish this.
The tea is ready, actually it's boiling.
Until you get the enhanced volcano, you don't leave this room. Did you guess this? I guess you did, right? I mean, read what I wrote before in the paragraph before. It's explained. It's there. There's no magic here. What about the shrinkage? We did it, right? You have it here.
I got the results for cancer.
Don't copy this because this is wrong. Okay.
So I got the 21224.
You got what?
No?
You should get a plot out of this if you run this. It's the answer of the universe. I thought it was 42.
Nobody gets the plot.
I think I'm going to have to change this. Sorry. Do you get something like this?
Wait. Now I'm getting it.
No. You should know by now.
So mine is running the plot.
So there's a different way to access the results. So these are the coefficients of all changes already pre-calculated. They are stored in these using these names. So if you want also quick access and the comparison is already pre-calculated, you can use the full name instead of this long contrast. But not all of them are defined, and I'll explain that maybe if I have time later. But yeah, to resolve the exercise that we're asking, you needed to fill this line and this, and it would plot a enhanced volcano. Any questions? If you read, the answer is there. I'm not, don't follow the screen. You pretty much have explained how to do this.
We have done actually.
The above line you don't figure out.
We've done it before. This is Rickage. I mean, you don't have even to do this. You could skip the second line and still you're going to get a similar volcano plot. It's not going to be that different. So if you notice, our favorite gene is still there as one as being highly differentially expressed, right? Positively also. So within the knockout, Plin 4 is still important between sleep deprived and non-sleep deprived. So if we plot that gene, and we separate the data between the 4 conditions. What you can tell, right? So PLIN4 is important for the sleep deprivation, but it doesn't matter if it's knockout or not. If it's a knockout, the gene still is going to create big difference within normal and within the knockout.
What is third? What is third? Fourth, shan't normal and
Oh,
shan't.
what is missing?
Shan't is sleep deprived. Shan't is sleep deprived.
No, normal, right? Oh no, normal it is here. Sorry, which one is?
Shan't three normal.
You're deprived. Sleep deprived. Yeah, exactly. The sleep deprivation in shack 3.
The shan't is not playing much role.
नहीं, it is not being displayed, but ऊपर है ना.
No, मैं समझ गई, but it's not because the sleep deprived is also coming.
This gene is barely affected by the genotype. They don't care what genotype it is. It's only affected by being sleep deprivation. So before we have T, one thing that is really important, citing, I guess, if we're going to cite the paper, but this line here, let's print it and you see what it does. So it's quite common in Bioconductor and maybe other packages that if you request the citation of a package, they generally generate the citation for you so you can cite it in your publications. But this one is quite important too. The session info, what it's doing is describing all the packages that you use and what version was used, right? So if you need to come back and somebody tells you, oh, what DSIC version did you use? You already printed, you save it, it's in your document, so you only need to check it. Why we put it at the end? Why we print the session info at the end of the Jupyter notebook?
So like everything you do can be captured basically?
Yep, that's the idea.
For the reproducibility of further.
Yeah, definitely, but we do it at the end, as he said, because let's imagine we load the libraries at the beginning, but we could have load the library in the middle of the code, right? So we want to wait until the end to print the session info. If I tell you the truth, I barely do that because I always load my libraries in the top, but it's always a good practice to put it at the very end. And the notebook is done, and you can have tea. Questions? Questions, problems?
Guys, speak please. Maybe whosoever wish to have tea can go out and have tea. Because they close the tea counter by 4:30.
So just one more thing that I didn't explain. So we are asking that column, right, in our condition table to compare this versus this. A positive fold change, sorry, how to say that? This is the numerator and this is the denominator, right? So the first factor, level of factor you're given is going to be the denominator, numerator, and this is the numerator. What is the one on the top?
Top is numerator.
Oh, top and bottom. So a positive fold change is going to be something highly expressed here. Sorry, I didn't explain that.
We do suppose the disease conditions, the divided by the
Normal, yes. So this is the idea. So disease normal. So I didn't explain that.
Up for this, it will be down for that, one will be down for that, one will be up for this.
So if you want the opposite, you change them.
No, no, no. Suppose ये मालूम मैंने गलती से कर भी दिया, पर अब मुझे पता है कि मेरा जो डाउन है ना कि मेरा एक्टिविटीज इन्फॉर्मेशन में आप तो अगर जीन्स पर डाउन रेगुलेटरी टू चेंज करने के लिए हाँ आपने क्या
Can I? was looking for more questions. That we need 3 pieces of information to run this SIG2, right? We need our count matrix, row counts, that's really important. We need the row counts because there's going to be a transformation, a normalization process. So if you provide something normalized, re-normalize something, it doesn't make any sense. Our metadata that can be, for now we only work with one column, but we can have multiple columns, and that should be the topic. I don't know if we're going to work with this tomorrow or not, or we close the topic now. But we can have multiple columns and work with multiple factors. But we just work with one today, right? We start with a condition. We was comparing normal with the sleep deprived. Then we use a different column, but still it was only one column that had 4 factors. So when we give the design, what we're asking is column names of our metadata, right? So we want to study, is the condition the main source? So we're saying the expression differences we observe are based on the condition. So, and the SIG2, oh, I have this. right? We provide the 3 pieces of information, and what we were testing is that, right? So if this difference was significant or not, different from 0 or not. And this was actually picked the gene with the highest difference in this case, because it was pretty obvious. So we think about it, what we provided this design. We've seen the output table. I guess I was a little bit fast, but I'm going to repeat again. So this is just the average across all samples, the log to fold change, the standard error of the fold to p-value and p-adjusted value. Questions about this? The problem came when we had multiple factors within a condition, right? And then in those cases, what we did is to provide the contrast to access the other pairwise comparisons. So the function of contrast is to access the other pairwise comparisons. If you don't use contrast, what DSeq2 is going to do is to find the first comparison and give you just one. When you ask for the results, it just gives you one pairwise comparison, not all of them. So the way to access it is to tell me, we tell it, please, from the column that I'm working with, compare this with this. That's what we said, right? So that's a way to access other comparisons that are not present by default. Because DSeq2 only returns one table at a time. So if we have multiple treatments, imagine we have treatment 1, 2, and 3, and 4, you just keep it in a single column too, and you need to do pairwise comparisons. That could be an option to do the analysis, right? So I show you the model matrix. I explain quickly, again, for this type of comparisons, even if more complex, you don't need to access the model matrix, but you could, right? So this is how it's aligned. Basically, it's telling you the grouping that we're going to be using and how internally it calculates the coefficients. So to calculate the baseline, it's only going to use the normal samples, right? So it doesn't use the condition slip. So it calculates the baseline. Sorry. Oops. Calculates the baseline. If we could plot an arrow from here to the baseline, and that would be the coefficient or the baseline or the intercept. And the other one has the baseline plus something else, and we are estimating this. That's how internally it's making the calculations. And I'm trying to plot this because if we do, that's what I was saying, if we do more complex models, it's good to visualize to understand what coefficients this is calculating. Most of the time, you don't need to do this because it's easy to understand just looking at the formula. But I don't know what you're going to be working with. If you start working with 3 or 4 factors, each one of them has 4 or 5 levels, it gets really complicated. What factor or what coefficients DSeq2 is using or calculating. So we give you this example, right, yesterday. And thanks to the PCA, we observed that, you remember that example I gave you the PCA with the mice in cages? There was this strain, there were...
So if comparing multiple factors, how is the coefficient, will the coefficient be different for each comparison, or it will take a mean of everything?
No, so what DSeq2, let's imagine we give them this formula, right? So we're using different factors here. We're using the strain, the gender, and the condition. For simplicity, let's assume there's only 2 of them in each one of them, right? So we have only 2 genotypes. We only have 2 genders, I guess. And we can have 2 conditions. So DSeq2 is calculating only 3 coefficients. No, it's not doing anything else. It's calculating 1 for condition, 1 for gender, and 1 for strain. And we can talk about this next, I think. OK. So somebody was asking about batch effects. So when you work with batch effects, then is when you start getting more complex formulas, right? Let's imagine this situation. We have a condition, normal versus a KO. You have this situation, but you realize that within each of these conditions were analyzed by 2 different labs. and you suspect there could be a batch effect. Actually, when you plot the same thing and you separate by lab, you notice that there's some sort of batch effect here. You see that the condition changes are similar, but they're at different levels. So this is a classical batch effect situation, right? Your baseline, your normal, is different from lab A than from lab B. If we analyze this, there's a lot of noise here. We may be missing genes, right, that could be differentially expressed, significantly differentially expressed because of the noise generated by the lab. That's a batch effect. So there are ways to deal with it. You can use tools to remove batch effects, right, which would modify your data. So you can use Lima, which is another R packages that can deal with batch effects. Or you can model it, right? You can add it to your model. What I recommend, I don't know, I prefer the model idea. For whatever reason, I don't know. I mean, the word batch, you can define it in different ways. We think batch effects something technical, right? Which is the case here. The lab is not something that we care. It's not part of our question. We kind of want to remove it. There's a preference about modeling or removing. I don't know. I like the idea of modeling better than removing, but I don't see a problem either, right? You can use Lima to remove the batch effect and keep doing the analysis. If you want to be working with DSeq2, it seems easier to just model it. It allows for that, right? So actually we have, so this blue line, as you can see, is the actual effect we want to study, but there's some effect also caused by the lab. Yep. Yeah, and this is what we generally call batch effects, right? But I mean, I don't know, there's also biological effects that can batches, right? We have the batch of being normal and the batch of being KO. But generally, when we think of batch effects, it's about technical issues. So now we have a new metadata, right? So we have two columns. So we could formulate it like this, right? So our expression depends on the batch and the condition, because we notice that it has an effect. So if we do the model matrix, now, again, we have an extra column. We can see the classification between them. So now it changes a bit. We have a new baseline, right? So the new baseline is the normal one based on how we build it. We can build it differently. The baseline could be different, but the way we have it here in the table, the one that has zeros everywhere except for intercept is normal lab one. So our baseline is this. Yep. So now, this is what DSeq2 is going to do. The first coefficient is going to calculate is the next one, which is the one for batch lab B, based on this thing, right? So the one that only has a 1 here and 0 everywhere else are these samples here. So it's calculating, okay, this is a coefficient I'm going to calculate. So from the baseline to the batch effect of being normally lab B. Now, the next coefficient I'm going to calculate is this one here. The only one that has zeros everywhere and only ones here is this one. So this is the effect of the condition. So that is more or less how internal is going to work. I know you don't care about these things, because basically what we're saying here is give me two coefficients, one for the condition and one for the batch. I only care about the condition. So I'm only going to ask you, give me that coefficient. And by default, if you build the design formula like this, DSeq2 only returns condition. If you ask for the result table, it's only going to return your condition, not the batch. So what you're doing effectively is removing the batch effect to some extent. If you don't ask for it, if you don't use contrast, you're not going to get it. So the way to get the red line, I don't know if you care, but if you care, you need to do contrast, batch, compare this versus this, and it will return you the red coefficient here. But if not, it's only going to return blue. That's by default how DSeq2 works. So if you don't want to think about how these things are built, basically the question you're asking with this formula is, what are the difference between KO and normal regardless batch? That's what you're asking. Yep. So if I switch batch by condition, if I say condition plus batch, DSIG2 by default will return the red one and not the blue. So when you build the formula, if you want to deal with multiple things, put at the end what you care. So if we have, this can be as complex, I think I'm giving an example more complex than that. Well, like, here is what I'm saying. By default, a DSIG2 is going to print only this. So I'm giving an example here. So if we ask for the coefficients, they are all calculated, right? If you ask for that table we saw before, the coefficients table, you're going to obtain all the coefficients. But if you ask for the results, you only get the ones, the condition ones. So if you really want to access what is the effect of the batch, you need to use the contrast to ask. If not, you don't get it. Yep. So, oh, you can, it's not going to show up, right? Because we have the problem. But I mean, I guess I can ask this. If we want, it's showing up, yeah, but it doesn't show live here because it's asking, the presentation is not live for whatever reason. I need to do that trick. I mean, we can do it. I mean, we can try. Let's see. Oh, no, my bad. I actually need to start it. So can you build a design formula to answer this? So build a design formula in a way that my question that I'm trying to answer is, regardless of the genotype, I don't care if it's normal or SHANK3, I want to know the effect of being sleep deprived or the effect of condition. So regardless of genotype, I want the condition. Based on what just tell you, what is the design? Can you, don't be afraid of typing, but don't use white spaces every in a single word.
Baseline is the normal here.
Don't worry about the baseline. You don't need to know what's the baseline. But yes, I mean, if I do the level thing, I'm going to say that this is the baseline. And this is the baseline for genotype, and this is the baseline for condition. So each column can have their own baseline. Or, well, if you think about with the graphic that I show you, it depends on how you build the formula again. So my question is, what is the effect of condition regardless of the genotype? Remove the genotype effect and just give me the effect of the being sleep deprived. How is the formula you're going to be using? Is people typing? Are you guys typing things? Even if it's wrong, just type something and see if it's live.
So it's not updating.
It's not updating? Nothing. Well, if it's not updating, we can keep going. I'll give you the answer. So if your question was that, right? So I only care about the difference in the condition. I don't care about the genotype. And I want to remove the effect of the genotype. As I told you, put the last element of your formula, the one that you care. So I care about the condition. I don't care about the genotype. And by default, DSIG2 will return the effect of the condition and not the genotype. If I only use one column, right, I get mixed up these effects. So I'm not calculating the genotype effect. If I use both, I'm splitting the variance on the data, right, into two factors.
Why we need to add genotype? We will give only design is equal to metadata and condition.
Okay, so if you only use the condition column, you are mixing the genotype effect in your formula. So if I remove this, right, and I'm only using a column, the genotype effect is going to be mixed up with the condition because I'm not giving this information. If I want to separate both and calculate both, I can use this formulation and keep them separated, but I'm only reporting the one that I care. Yes? I don't know. Your faces doesn't look like you. I'm not doing a good job. I don't know if there's a different way to explain it. Verbally is the best way that I found to explain these things. When you build your formula.
Kept is constant. Genotype is now constant.
Genotype is constant. No.
But it's not accounting, like.
So it's this problem we had before, right? If I go backwards.
What I mainly want to see, that is coming at the end. I want to see the effect of the treatment that I put at the end.
Yes.
BSeq will take that as a primary motive to, analyze the data. But
So
at the same time, I don't want to miss out the effect of the genotype. So
because if you don't, you have this. This situation is the same one as the genotype, but if you call genotype A, genotype B, you are in the same situation, right? So you prefer this situation where you clearly can separate the effect of genotype and condition than working with only one column without not knowing what the genotype is doing, right? So that's what I'm trying to explain. So the batch effect situation is identical that the other one that we talk here, right? It's exactly the same thing. The only difference is I call it with a different name, but this could call it batch.
So if you actually take a moment away and then from mathematics of it, think about what is the biological question you are trying to address here. What is the biological question you are addressing?
treated versus non-treated.
No, just tell me in one sentence what is the biological question here. We are addressing here that if SHANK3 knockouts behaves differently in wild type, sleep deprived and normal, if there is any difference or effect of SHANK3 mutation in sleep deprivation.
But hold on. No. So hold on. Your question comes later on. The question I'm trying to answer here is, what is the effect of the condition regardless of the genotype?
No, that is fine. But the issue here is that in the data, there are two components of the data. One is the genotype, which is loss and gain of SHANK3. And we understand that anything what you do with the rest of it, sleep deprivation, otherwise, there might be some effect. That is the null hypothesis. So genotype needs to be taken into account in your model, unless you will not get that. Because that is the null hypothesis. And if you want to really to see that null hypothesis or accept or reject the gene without the genotype in your equation, it will not. You will not be able to see that.
Yep.
So what really will happen?
Yeah, use it.
I want to use it.
Yeah.
No. So if you are actually just using condition, then what will happen? The genotype will confound your.
Yeah.
No, I want to see that also. I
Yeah.
put the condition in my experimental design. I don't want to miss out on my genotype as well.
Oh, sure. I mean, you don't miss it if you do this, because you use contrast and you get it. What I'm trying to say is, I'm just hypothesizing if your question is the one on top, how you would formulate it. I know, and you can change this word by batch, you can change whatever. Yep. I think you got it now, right? I think we got in a better position. So I think it's good if you formulate your question in paper to understand how you do the formula. And
That means we should stop for so that we can set up.
yeah, so because it's going the next question is a bit different, but I think we can leave it here.
We'll continue tomorrow also.
Yep.
And there is a suggestion, and it all depends upon all of you. So the suggestion here is we have the multivariate analysis also, the multi-factor analysis was also there, which we can discuss some more if you want. So if you want to spend some more time tomorrow, we can start early and end late.
We can do half an hour tomorrow. We finish this and then we move on.
Like if you want to start early, like maybe 8 o'clock, but everybody.
8:30.
And yeah, but then 8:30 means 8:30. Everybody should be here at 8. No, we start at 8. So,
and we start at 8:30 because today was 9 when we started.
Then actually you want to stay a little later also, then we can continue, because if you want to go through Friday is the only day we are left with variant analysis, so that means our RNSC, single cell bulk, all that discussion needs to be completed by tomorrow. So it's all about, like, if you guys are ready to work,
or a little harder tomorrow, then one day, a little more effort, then fine. Otherwise, it's okay.
I know. So at least we're going to do an exercise about formulating questions and how you put it in a formula. So I'm going to try to forget a little bit. I know this sounds, drawing lines like this, it sounds weird now. I'm happy to explain it better, but I think phrasing helps a lot too. And knowing what DSIG2 does with that formula.
Translate. Can you introduce Rick?
No.
Please do that.
No.
He's your friend.
Nope.
Given some churros, he will then introduce.
No. We agree.
Can you hear us? Sorry.
You're muted, Tony.
I'm muted. Really? Can you hear me?
I can hear you. Can you hear me?
Yes, yes, I can hear you.
Excellent.
Can you hear me?
I can, yes.
Okay, great, great. Okay, so let's